صباح الخير. تسمعني أستاذ أحمد؟ صوت واضح؟
نعم تفضل.
شكرا جزيلا. Very good morning to you all. I am Asma, Head of Scholarship and Cultural Relations Department. I am the Master of Ceremony for this morning conference proceedings. Facing unknown is very difficult. In the face of an epidemic, we are uncertain how to respond, threatened by contagion, terrified by the unknown risks. We move into survival mode. We worry. We ruminate. We have the urge to connect and the fear of contamination. We want to lock the door, but fear isolation. We become saturated with media warnings and shaken by the shreds of frightening information and even conspiracy theory. But nothing stands in the face of a humanity, and life goes on. Highly respected scholars, researchers, and participants, Assalamu alaikum wa rahmatullahi wa barakatuh. A very good morning to you all. With the greatest pleasure and on behalf of the University of Al-Qadisiyah College of Biotechnology, I would like to welcome you to the first international virtual conference of biotechnology here in the city of Diwaniyah, Governorate of Al-Qadisiyah. We are very grateful and glad you could make it. Special thanks and appreciation to the President of the University of Al-Qadisiyah, Professor Dr. Kazim Jabr Al-Jaburi, and to the Dean of the College of Biotechnology, Assistant Professor Mohammed Al-Askari, and special thanks and appreciation to the members of the conference committees for their tremendous efforts exerted to host, organize, and put together such an engaging and decisive research topics. I sincerely hope you will enjoy attending the conference sessions, and thank you for your participation. Let me very briefly take you through this morning opening ceremony program, which will start with the national anthem, the recitation from the Holy Quran, then followed by a welcoming speech by the President of the University of Al-Qadisiyah, and shortly after the Dean of the College of Biotechnology and the Chair of the conference will deliver his speech and announce the start of the key session. Now we'll start with the national anthem. النشيد الوطني أستاذ أحمد.
موطني الجلال والجمال والسناء والبهاء في رضاه والحياة والنجاة والهناء والرجاء في هواه هل أرى سالما منعما وغانما مكرما سالما منعما وغانما مكرما هل أرى في علاه تبلغ السنا موطني الشباب لن يكل همه أن يستقل أو يميل نستقي من الردى ولن نكون للعيب كالعبيد لا نريد ذلنا المعبد وعيشنا المنكد ذلنا المعبد
Thank you very much. And now recitation from the Holy Quran تلاوة معطرة مباركة من آية الذكر الحكيم. تفضل دكتور أحمد. دكتور أحمد.
بسم الله الرحمن الرحيم اقرأ باسم ربك الذي خلق خلق الإنسان من علق اقرأ وربك الأكرم الذي علم بالقلم علم الإنسان ما لم يعلم كلا إن الإنسان ليطغى استغنى إن إلى عبدا إذا صلى أرأيت إن كان على الهدى أو أمر بالتقوى أرأيت إن كذب وتولى ألم يعلم بأن الله يرى كلا لئن لم ينته لنسفعا بالناصية ناصية كاذبة خاطئة فليدع ناديه سندع الزبانية كلا لا واسجد واقترب
صدق الله العلي العظيم. And now I would like to welcome the president of the University of Al-Qadisiyah, Professor Dr. Kazem Al-Juhuri, to give his welcome address in Arabic, followed by a just translation to English. دكتور كاظم تفضلوا. كلمة دكتور كاظم. جاهز دكتور أحمد؟
مع حفظ الألقاب العلمية والمقامات. وأشكر من تطورات وأحداث عصفت بمعظم بلدان العالم. إن من دواعي سرورنا أن نلتقي معا في أعمال المؤتمر الافتراضي الدولي الأول الذي تقيمه كلية التقنيات الإحيائية في جامعة القادسية تحت الشعار ببحثنا العلمي نواجه جائحة كورونا المستجد للفترة من 29 إلى 30 حزيران. أرحب بكم جميعا أملا أن نجد وسائل جديدة ليصبح عالمنا أفضل وأكثر ترابطا، وأن نقدم لأفراد مجتمعاتنا كل ما يمكن أن يحمله المستقبل لهم من آمال وطموحات. أتمنى لكم النجاح والتوفيق في أعمال هذا المؤتمر، في قراراته وتوصياته، وحجم المسؤولية الكبيرة الملقاة على عاتقنا جميعا من أجل توفير حياة كريمة ملؤها الرخاء والازدهار. والسلام عليكم ورحمة الله وبركاته. الأستاذ الدكتور كاظم جبر الجبوري، رئيس جامعة القادسية.
Thank you, Doctor. A very warm welcome to all of our highly respected guests, participants, and scholars, and thank you for your contribution. Despite the difficult and most exceptional time the world is facing, we at the University of Al-Qadisiyah are pleased to host the first international virtual conference of biotechnology held by the College of Biotechnology, and under the slogan, "With our scientific research, we face the emerging coronavirus," for 2 days, 29th and 30th of June. I would like to welcome all of you in the hope that we all can come together and wish you all the success and the proceedings of the conference, and all the best with the drafting of decisions and recommendations that will directly be related to the development of well-being to enhance the quality of life. Thank you, Doctor. And now the word to the Dean of the College of Biotechnology and the Chair of the Conference, Assistant Professor, Dr. Mohammed Al-Askari, to deliver his speech and announce the start of the key session. Tafadhal, Doctor.
Thank you, Dr. Asma. Assalamu alaikum. We welcome researchers, participants, and supporters of our first virtual International Conference of Biotechnology. Under the slogan, "Our scientific research challenges the COVID-19 pandemic to exchange ideas and scientific research results to save the societies." You are all most welcome in this gorgeous scientific room. Thank you. And hereby, I do announce the starting of our conference keynote session. Thank you once again, Mohammed Abdul Wahab Al-Askari, Dean of the College of Biotechnology at the University of Al-Qadisiyah.
Thank you, Dr. Mohammed. Now I will leave you with a short video about the College of Biotechnology. دكتور أحمد الفيديو لطفاً.
السلام عليكم جميعاً، ونرحب بالسادة الباحثين والمشاركين والداعمين لمؤتمرنا الدولي الافتراضي الأول في التقنيات الإحيائية، وتحت شعار ببحثنا العلمي نواجه جائحة كورونا المستجدة، حيث المناخ العلمي لتبادل الأفكار ونتائج البحوث العلمية الرصينة لخدمة المجتمعات. أهلا وسهلا بكم في هذا المحفل العلمي البهي، وشكرا لكم جميعا. محمد عبد الوهاب العسكري، عميد كلية التقنيات الإحيائية في جامعة القادسية. Assalamu alaikum. We welcome the researchers, participants and supporters of our first virtual international conference of biotechnology under the slogan "Our Scientific Research Challenges the COVID-19 Pandemic" to exchange the ideas and scientific research results to serve the societies. Once again, you are most welcome in this gorgeous scientific forum. Thank you. محمد عبد الوهاب العسكري, Dean of the College of Biotechnology, University of Al-Qadisiyah.
الشمال الغالي يبقى شامخ عالي يسموك الجبالي كاكا تمام والوسط قريبة والغربية حبيبة والفرح تجيبه باسم العراق كل العراق والجنوب يوافي والنخل عوافي ويا قلبها الصافي نبض العراق بيت كل اهلنا وصوتنا واملنا بصوت عالي كله نريد السلام كل زاهية وحبوبة ونادرة وطيوبة فيها بكل عذوبة هالقلبها سالمة يا بلادي يا تراث أجدادي ويا رمز بغدادي وعاش العراق يسحر النواظر ويسر الخواطر بين ماضي وحاضر دار السلام يا بلادي سيري مصيرك مصيري والوفا ضميري لأهل العراق عراق عراق كل العراق
Thank you very much. I hope you enjoyed watching the amazing video. It is undoubtedly that scientific research is the backbone of economics and development and is an important pillar of human knowledge. Therefore, and on your behalf, I would like to welcome our first speaker, Professor Kadhim Mohammed Al-Sumaydai from Al Nahrain University, Biotechnology College, a researcher in plant biotechnology. تفضل دكتور كاظم موجود ويانا. دكتور كاظم, can you hear me?
نعم معكم. نعم
صباح الخير دكتور.
الأنوار. شكرا جزيلا شكرا جزيلا.
أهلا وسهلا, تفضل دكتور.
اعملي لي السكرين رجاء.
نعم.
السكرين على المنهاج على
نعم.
بس حاولوا زيلوها حتى اقدر اساعد الداتا شو.
تمام. دكتور أحمد. تفضل دكتور, أعتقد إليك الآن. راح تسوي شير من يمك دكتور مو؟ إي نعم هسه ظاهرة.
نعم.
تفضل دكتور.
الحضور الكريم, متمنيا لكم الصحة والسلامة تحت هذه الظروف الصحية التي يمر بها بلدنا والعالم أجمع. شكري إلى جامعة القادسية من رئاسة الجامعة وإدارة الجامعة عموما وبقية التقنيات الأحيائية، ونسأل الله التوفيق إلى أخي العزيز الدكتور محمد العسكري وإلى اللجنتين العلمية والتحضيرية. My sincere thanks to the University of Qadisiyah, College of Biotechnology, and certainly the organizing committees. I'm going to talk on the recent advances in plant biotechnology this morning. First, we have to distinguish between two terms that are commonly used in agriculture and plant biotechnology. The conventional agriculture, which deals with the horizontal expansion of agriculture by expanding the land, reclamation of the land, and improving the quantity and quality of agricultural products. While the biotechnology concept is mainly concerns with the horizontal, with the vertical expansion. In other words, we are dealing with the better quality and increasing the quantity of a crop. Back to the past, if we look at this slide, please, this presentation, you look that the farming began in Mesopotamia many years before the BBC. So the people in Iraq, in Mesopotamia, have started the real agriculture. As the time advanced to 1990, the past century, the discovery of Mendelian laws, rediscovery, in fact, of the Mendelian laws, there is a revolution in plant biotechnology occurred during the last century. I'm not going to go to the details of these dates, but there are remarkable dates in this case. When the DNA was discovered, then the most remarkable point in this history of plant biotechnology is the production of GM crops, which grown in about 65 million hectares in 18 countries in 2000. And I don't miss to mention that the Babylon, just before 3000 BC, also discovered the beginning of plant biotechnology when they recognized that dates do require pollination. Looking at the world in general, so in this slide, please, you look and you see carefully that many countries, especially in Africa, they do have a deficiency in required daily calorie intake. In other words, in food supply. For example, Mozambique, there is a deficiency of 30%, Somalia 25%, Haiti 15%. However, the developed countries, they do have a surplus of required daily calorie intake. So now we are, as a biotechnologist, what do we have to do? So we have to make sufficient supply of food with better quality so we can make self-sufficient in these countries, including Iraq, rather than depending on the developed countries to supply us with food. When we go to the applications of plant tissue culture as just one branch of plant biotechnology, so if we start from the above, so we can use mixed plant culture, for example, for the micropropagation and production of millions of plants within very short time under controlled conditions. A bit to the right, if you look at the mycelia culture, so which is the upper side of the ovary, which contains 3 sets of chromosomes. So we could produce, for example, triploid plants, and in some cases, when there is a somatic tissue, we can produce diploid, which we call clonal propagation. On the other side, callus culture can produce somatic embryos, which I will go in detail later on. So this is a good source for vegetative multiplication and production of synthetic seeds. A little down, please, if you look at the anther and pollen culture, which is a very optimistic field in plant biotechnology in producing homozygous lines for plant breeding. So we could produce plants with one set of chromosomes, which are homozygous and can be doubled by using colchicine. Then we can convert these plants to fertile plants. At the bottom of the slide, the protoplast technology, which is really a revolution in plant biotechnology. So it's possible to isolate protoplasts from single cells or group of cells and then align or fuse two protoplasts with desirable characteristics and then production of plants with novel traits. Again, the endosperm culture, which is the starch or the store food in the seed, which is normally 3N. So we could produce triploid plants with bigger fruits. Again, to the embryo rescue, so it's possible in some process that the embryo will abort. In this case, the plant biotechnologist will interfere and recover this embryo and thus obtain embryo and then regenerate to a whole plant, which is novel, not exist before in the nature. And finally, the medicine culture, which can be grown, dissected and grown in vitro and produces a virus-free plants, which is very important in the agricultural sector. So these, if you look at the slide, please, these are the stages of date palm production, starting from callus culture, then induction of somatic embryos, then regeneration of whole plants, and finally cultivation in the field. Another application of plant tissue culture in plant biotechnology, that the hairy root culture. These explants can be transformed using Agrobacterium rhizogenesis, and then the proliferation of hairy roots will be a very good source for pharmaceuticals in general and secondary metabolites. Dyes are very important in industry, especially in clothing industry. So these can also be produced by tissue culture. The lower part shows that it is possible to produce millions of plants from a single explant. So it is really, the plant tissue culture can contribute significantly in the revolution of plant biotechnology. Protoplast fusion, if you look at these machines, these are rather cheap and can fuse two cells. I mean, protoplast is a cell, a normal cell, that the cell will be degraded by enzymes or mechanical process. So these protoplasts can be fused, and the novel product contains very desirable characteristics and also combats the incompatibility between plant species. These mutations have produced many plant chimera, which are very significant for ornamental purposes and for improving the quality to the consumer. Now, about the deforestation and plantation of wide areas. If you look at this airplane, this is an airplane bumper which was used to be used in the First and Second World War. So these have been exploited in bombarding what we call it seed bombs. So these have been converted, as you will see later on. So these missiles contain seed bombs. Seed, not in the real meaning of the seed, the normal seed, but it is produced by biotechnology, which contains somatic embryos being developed at a specific procedure and being used for agriculture. So each missile or bomb contains many capsules. These can hit the ground of the soil, especially in the mountain area and desert. So in this case, we could fully utilize the desert and the very steep mountains to grow plants and then improve the environment in general. Just to remind my dear audience that if Iraq has forested the desert and the steep mountains, the temperature will be reduced up to 7 Celsius, and this will reduce the cost of electricity and the reduction in using cooling equipments. So in this case, this bumper or airline bumper, this will equals to 3,000 workers. So you could imagine how we can reduce the cost of labor by using the new technology which has been established in many countries all over the world. So this is the seed, and if you look at these seedlings or plantlets produced in tissue culture, these can be even used for landscaping the urban cities and the rural cities alike. So these some pictures showing the process of growing the plants in such areas. The application of nanotechnology in agriculture has been enforced, especially in the last decade. I am not going to go in details on this slide, but I would like to mention that nanotechnology has been introduced strongly in the agriculture, especially in the plant nutrition, where nutrients can be carried or converted on nanoparticles and delivered to plant roots, and also in environmental sciences, especially for the biodegradation of pesticides and other pollutants in the nature. Also in the animal production and animal husbandry, and producing good quality food stuff to the animals. And finally, in the precision agriculture, especially in manufacturing nano barcodes and quantum bells, and also wireless nanosensors and other materials. So it's a very optimistic field in agriculture. Now, if you look at this slide, please, you look at the plant biotechnology has already pushed the world towards a very steady steps. So the improving the nutritional value in many crops, especially in golden rice and cauliflower, when some genes from daffodil plant being transferred to these crops, which contain carotenoids or provitamin A, so improve the quality and enriched these crops with vitamin A and prevented blindness in millions of children, especially in the East of Asia countries. The other application is phytoremediation. So the concept of phytoremediation, which means that we use plants for remediating land from contaminants instead of using conventional methods. So the concept is relied mainly on the cytochrome harboring P450. This in my lab, in another labs, also been that the cytochrome gene been isolated from rabbit because the rabbit is the most tolerant animal to the contaminants and being transferred to many plants, local plants which are grown in Iraq. So the future is optimistic that growing some plants which been genetically modified can reclaim or rehabilitate or absorb the contaminants from soil, not just soil, but air and water and groundwater as well. Another application is the production of plants resistant or tolerant to biotic and abiotic stresses. So it's possible through tissue culture to produce such plants, especially salinity. Since salinity threatens the whole world, about 1/4 of the irrigated land is now out of use because of salinity. So we have developed some varieties which are tolerant to salinity, and it is possible using genetic engineering to transfer genes from tolerant plants to sensitive plants. Also the case would be drawn on increasing tolerance to other biotic and abiotic stresses such as herbicides, drought, cold, flooding, and other stresses. The other application which I have already mentioned is the induction of somatic embryos, encapsulation, coating with fertilizers, pesticides, herbicides, and then fluid drilling. Fluid drilling means using machinery. So we are enlarged this seed, artificial seed, to be used for fluid drilling in the machines. And these seeds are, in fact, will achieve large scale production and true to type because they are somatic, uniform, and cheap, and reduction in cost. Bioreactors are used now for this purpose. This is the process of synthetic seed processing. So the embryos are the heart shape or torpedo shape can be embedded in hydrogel, then coated with the nutrients and used for this purpose. And this is the final shape after coating of the seeds which you have already seen, which have been bombarded by airplanes. Embryo rescue technique and in vitro fertilization is also a very optimistic technique in plant biotechnology. In many cases that the embryo will be aborted. So just before abortion, it's possible to manipulate, to dissect this embryo and transfer it to a new nutrient medium rich with the nutrients, and then we could get novel crops never been exist in nature. From the bioenergy side, so how can plant biotechnology contribute to bioenergy? Looking back at the past centuries, that the man depended on the water power, then steam engines, the railroad, and in just the past few decades, petrochemicals is the main, especially after discovery of oil. So the petrochemical era was the most common. Later on, starting from 1980 until 2030, it is what we call the information age. It is the age of obtaining information, digging in the literature, digging in the scientific research. So we are prepared as a biotechnologist for the next generation or next age, which is biological age, especially when oil depletes. So we are confined to look at the biological age, we are then considering the biological sources, especially plants, are the main source of energy for a human being. If we look at the slide, please, I'm not going to talk about the first generation and second generation of biofuel production, because the first generation was dependent on the extraction of seeds and then extracting the oil or sugars from these seeds, which in fact made imbalance in the national trade and international trade on the complaints of the consumers. Then the second generation to the residues of plants, which also affected the animal kingdom, which consume these residues. Now the third generation of biofuel depends on the algae, particularly on microalgae. So producing these in these glass bioreactors can produce biofuel as a bioethanol, biodiesel, and other petrochemical derivatives. And this is a quite delicate land. This is a neglected land in America being fully utilized by bioreactors. These are not solar energy plays. These are not solar cells, but they are bioreactors for growing microalgae. And if you look carefully, you could see even the refinery is inside or in the middle. So there is no need for transportation, and each county can produce its oil derivatives or oil petrochemicals. At the home scale, it's possible to produce biofuel, bioethanol for your car as well in an area that does not exceed 1 square meter. And also these bioreactors for producing biofuel being installed in national parks for ornamental and landscaping in addition to the biofuel production. Now we are turning to another topic, please. If you look at this tree, please, this is the crown gall, which is a serious disease in many trees. So the reason for this disease is a kind of bacterium, which is called Agrobacterium tumefaciens. This bacterium has been discovered is really a genetic engineer. We call it a genetic engineer because it has the ability to confer a gene from any organism and convert then transfer to the plant to improve it. Genetic engineering is not only confined to this bacterium by using in vitro methods. As a field, this gene bombardment can be used at the field stage. So the DNA particles can be loaded with golden particles or gold coated or even tungsten and being shotted to the plant tissue, then transformation will occur. And after that, this leaf will be dissected and regenerated to all plant. Then we look at the transformants. And this is at the lab scale. This is a gene gun for the transformation of plant tissue at the lab scale. The products of biotechnology are many, including, for example, seedless watermelon and corn or maize, which is very rich with antioxidants. And even we do need, in some cases, large fruits for the, especially for restaurants and the pupil consumption. So these just examples of biotechnology products being produced using the biotechnological concepts. The situation is not confined to plant biotechnology. Also, also for the animal production. So it's possible to transfer genes for wool and production of sheep with high quality wool and very woolly. So these can be used for industry and also for ornaments for producing horses with chimera and even production of supercars and super animals and even mini animals. So this is the disease is insect which called is corn borer, which in fact makes a very serious damage to the corn plant. And this being combated by using a microorganism is a sort of bacteria which is called Bacillus thuringiensis, abbreviated by BT. So the gene from this Bacillus thuringiensis being introduced to the maize plant and even to other plants like sunflower and cotton, and these plants showed resistant to this this worm, this larva, this insect. So fortunately, the engineered plant has no harm on the human being or animals because it does work only when the stomach is alkaline. Now again, if we look at the plant cell can be described the invisible large evil factor in the world. So it's invisible, and it is in fact the largest factor in the world. Factor with no smoke and burning in water. It's really a miracle from the biotechnologist point of view. If we manipulate Robiscon enzymes, for example, is now as a good way to improve photosynthesis in the plants and then increasing the productivity, and ultimately the crop yield will increase and we will feed the people who are hungry all over the world. If we manipulate the cytochrome P450, this will absorb all the contaminants in the nature or the environment, so we can get rid of the pollution very cheaply. What expected from the next revolution? So the biotechnologists in fact really looked at why legumes can produce nodules for nitrogen fixation, and ultimately they do require very, very low levels of nutrients or fertilizers. So now the biotechnologists are thinking of manipulating leaf genes, nitrogen fixation genes, and manipulating these genes to the whole plant kingdom. In this case, a huge reduction in fertilizers will occur. This will be expected the next green revolution, and many researchers all over the world are working on the side. Molecular farming is also being introduced into plant biotechnology strongly. This is in fact will make a combination between genomics, proteomics, and bioinformatics. So this for the production of vaccines. In fact, almost about 150 vaccines or antigens being produced, the nutritional quality of food being improved dramatically, and hundreds of therapeutic products being produced. So in the near future, we expect there is no needles, no pain for children. Instead of that, consuming one strawberry or a part of banana or a part of apple will make the vaccination and do the job correctly after calculating the right dose. Production of pharmaceuticals in general from plants is quite a new branch of science. So this is being depended by the pharmaceutical companies and pharmaceutical industry in general by introducing DNA molecules to plant cells, and these will express the vaccine or the therapeutic protein or the therapeutic cure in many plants, then extraction and purification, then being encapsulated and sold in the pharmacies. I'm just about to finish. So perhaps some of you will think that this is a part of a scientific fiction. If we look carefully at the structure of the chlorophyll molecule, so the center of the molecule is the magnesium molecule, while the center of the human and animal blood is the ion. Is exactly the same structure, exactly the same structure. So what happens? The scientists now, and this is a breakthrough in the science of biotechnology, of transferring the ion molecule and replace the magnesium molecule with iron molecule. So the plants will produce blood. Probably from another point of view, if the vice versa occurs, that the magnesium would be inserted in the instead of iron, probably the human being will produce its food and energy and can be used as a source of carbohydrates, proteins, and lipids, and there is no need to eat food as at all. In biotechnology, we do need to preserve the genetic materials. Many laboratories all over the world, they do care of preservation of genetic material. So, in case of disasters or for any case, it's possible to preserve by cryopreservation of genetic material using any plant part. And this will remain being frozen in liquid nitrogen for millions of years. This is theoretically. So these are some of the gene banks which are available in many countries, and we do encourage the Iraqi government to establish a gene bank in Iraq for the conservation of genetic material in different parts of Iraq. And almost finally, we are looking for the story of jumping insect and the huge tree. This is a real story. Some of the biotechnologists looked at some insects which have the ability to jump tens of meters. So something around 1 centimeter can jump such a distance, 10 meters or more. So what is the secret in this insect? So some scientists, particularly in Sweden universities, in cooperation with Hebrew University, they were able to isolate these genes responsible for elasticity, and these being transferred to sequoia tree. Sequoia tree is the most or the biggest tree ever in the world. And the parts and the fibers and the cellulose of this tree is very flexible. So just making shoes, for example, for the military purposes, the soldier can jump tens of meters, and even for clothing. So one standard dress can be used because of the elasticity. And also the story of collagen, which is very well known to be used in plastic surgery. So instead of obtaining this source of collagen from the dead babies, the gene responsible for the production being transferred from babies to tobaccotry, and the product that plants produce about 6 times the production of collagen. This is the biotechnology tree in the future, as we expected to be. That's when a tree can compare animals, can compare different fruits on different plants. And mainly, for your curiosity, it is mainly produced by protoplast fusion. Thank you for your attention very much. I'm sorry for the
Thank you ever so much, Professor. Rather inspirational and deeply informative lecture. Thank you very much.
Thank you very
Thank you. And now I would like to call Dr. Bilal Al-Ubaidi, a researcher in medical science from Newcastle University. Human genomic, that's I think specialty in human genomic, genetic variation and genomic technology. دكتور بلال موجود ويانا دكتور بلال؟
نعم موجود نعم.
تفضلوا دكتور صباح الخير.
شكرا جزيلا صباح الخيرات.
أهلا وسهلا.
Will you let me share my slide?
Yeah, go on.
Okay. Can you see it?
Not yet.
Wait a second. Yes?
Yes, we can see it now.
Okay. بسم الله الرحمن الرحيم. شكرا جزيلا. First of all, I want to thank Al-Qadsiya University for hosting this event, and special thanks to my colleague, Dr. Mohammed Al-Askari, for inviting me to be one of the keynote speakers of this event, and I appreciate it. And I also want to thank the team behind the scene who are working really hard to make this event happen. I know it's, believe me, it's not like an easy job to organize and host these events, although it's like an online event, but there are too many logistics and stuff need to be done properly. I want to talk today about the human genome genetic variation and genomic technologies, and I might explain some of my slides rather in Arabic because of my audience are mostly from Iraq. So yeah, so the slides will be in English, but some explanation will go on in Arabic as well. Well, I first want to start with this, say, from a Nobel Prize winner, Sidney Brenner, just to explain the importance of the new technology. So it says, like, progress in science depends on new techniques, new discoveries, and new ideas, and probably in that order, just to mention that the importance of the new techniques that emerging every now and then in the world, and how these techniques are like helping to find new discoveries and apply the new ideas that come from different people around the world. So when we look at the genome of the human, we are actually looking at something like this picture. We're looking at 3 billion letter of ATCG. And the aim of looking at this huge amount of data is just to help in either diagnose diseases, as like this is the major interest in the medical field, and or like what the Dr. Kadhim, my professor, Dr. Kadhim, who talked first, was emphasizing on the importance of the improving the research in the plant technologies, and how this will help in providing food and protect the environment and so on. So many applications of these things nowadays. So the major challenge in medical genetics, which is the branch that I will be focusing on today, is to use these data for identifying genetic variation and interpreting genetic variation. And in research, large long-term studies to identify genetic variation that increases or decreases risk of disease, and functional studies to confirm the pathogenicity and reveal the mechanism. While in clinics, the diagnosis in individual patient and who want reliable and useful answer fast and affordable. لأنه طبعا احنا نركز على الجينيتيك ميوتيشن أو الفاريانتس كما نسميها. الأهداف يعني عديدة، منها مثلا في البحث العلمي نريد نعرف الأسباب، بينما في الكلينيك أو الدكتور بالمجال التطبيقي المريض يريد جواب لسؤاله شنو المشكلة اللي يعاني منها، ولذلك يعني البحوث تجري يعني شلون هاند تو هاند مثل ما يقولون، أو يعني تعاون بين الباحث والكلينيكس لغرض الوصول إلى أجوبة اللي يحتاجها المريض. تقدم يعني سريع صار في مجال الجينيتكس, and this is there's like too many breakthroughs that happened in genetics, and it's moved from sort of art in 1989, and now it's like kind of an industry. And we are talking here about the huge sequencing machines from Illumina that enables the sequencing of the whole genome from the human in few hours, actually. And also we'll talk later about the reduced in the cost that happened in the last few years. Everyone interested in genetics will remember, of course, that the first human genome project is started late in 90s and just finished in 2003, while these days, I mean, it's required about 13 years or so. So now it's required about mainly like with the analysis, and so it'll be less than 2 days in order to get the results. So our genome is full of variations. We are looking at 6 billion nucleotides per human genome, and every 2 people vary at 4 million positions apart from the identical twins, and 1 variation or mutation can cause rare disease. Single nucleotide variation can cause disease, but it's also too many different variations that indeed exist in the genome, and the genomic studies reveal that there is insertion or deletion of single nucleotide insertion, single nucleotide deletion or inversion, also deletion of DNA sequence, microsatellite repeats variation that my colleague Dr. Richard will go through in the next presentation. Also the genetic studies that reveal the existence of additional chromosome or chromosome rearrangement that keeps on happening every single division of the cells, or for some people. And variation per genome, so single nucleotide variation, these events that happen, it's like we're talking about 3 to 3.5 million SNVs, single nucleotide variants, of which vast majority are SNPs and single nucleotide polymorphism. And these are now there's a database to reveal these. And while the focus is on the 500 private or rare coding variants, and the 50 to 100 de novo mutation per genome. And mentioning de novo mutation is the mutation that exists in the individual that wasn't inherited from any of their parents. And indel, it's like insertion and deletion, the 300,000 indels, and largest uncertainty is still difficult to detect. And the copy number variation that also happens, it's like when we talk about copy number variation, I mean like the fragments of 100 base pair or till like about 10 megabase. And these are frequently happening in the genome, which is about 1,000 per genome, and more than 50 kb, there are about 30 times per genome. And for the de novo, it's more than 100 kb and less than 1 per genome. أنواع التغايرات هنا يعني دا أتكلم عنها اللي موجودة بالجينوم، قد تكون تغاير بقاعدة واحدة وهاي أعدادها يعني واضحة بالسلايد، قد يكون insertion أو deletion، وقد يكون النوع الآخر من التغاير هو الكوبي نمبر فاريشن. هذه الداتا طبعا ما كان ممكن الحصول عليها بالطرق التقليدية. Like this here is the importance of the next generation sequencing technologies and the huge effort of the bioinformatics, like that will start after the sequencing of the human genome. Detection of variation, all the solution from nucleotide to chromosome. So we have the old generation Sanger sequencing that we used to use, and these enable us to sequence a fragment of DNA with the size of 1 to 500, maximum 1,000 nucleotide. And then we also have the whole genome view and the karyotyping that helps us in the field of studying the chromosomal variation. And we also have like the microarray that would provide a huge amount of data for studying and confirming the results coming from the sequencing. And now as we'll talk more about next generation sequencing that enables to do the whole genome sequencing. So comparing between the traditional sequencing, the Sanger sequencing that we used to use, and that one is enable us to do like maximum of 96 DNA fragment, and that can be sequenced simultaneously, while the new technologies, and especially for Illumina that we are using so much, and will be like you're sequencing the millions of DNA fragments sequenced simultaneously. And it's also need to mention here that's enabled the sequencing or pooling of different samples together. يعني احنا سابقا بالتقنيات القديمة من نريد نسوي sequencing لازم ناخذ السيكونس من كائن معين ونعمل sequencing لقطعة معينة، وبعدين ممكن قطعة أخرى إلى أن نجمع أكبر قدر من المعلومات. الآن في التقنيات الحديثة اللي هي next generation sequencing نقدر يعني التقنيات في استخدام الإلومينا نقدر نسوي سيكونسينج ل 96 شخص كلهم في تيوب واحد، ونقدر نسوي whole exome sequencing ل 96 sample together at the same time. And also there are other like technologies that Oxford Nanopower and PacBio and Ion Torrent، هذه كلها يعني شركات أو تكنولوجيز مختلفة كلها تقدم نوع من السبورت للnext generation sequencing. هذه طبعا البدايات أو الأولد فيرجن من next generation sequencing devices. And these are from different companies. The one that now used most is Pacific Biosciences, PacBio, we call it PacBio. These are, and Illumina, the HiSeq, and even the more advanced version, which is NovaSeq. These are used for, like Illumina is for short reads and PacBio is for long read, as well as Oxford Nanopore that I will show you later. So here in our institute, we have these two devices. The one on the left is the most advanced the sequencing platform from Illumina. And on the right side, you can see the Oxford Nanopores or Minion, which is the size of the mobile phone or even smaller. And here to imagine the size, this is me and my colleague Giles. So on the left side, you can see the NovaSeq machine, and on the right side, you can see how small is the Oxford Nanopore machine. And you can imagine like this small device are able to do the whole genome sequencing for like a couple of samples at one run. And this is the Oxford Minion Nanopore. The development or the application of these different devices are different. I mean, you can see from left to right is the size of the fragment that can be sequenced using the different machine. And you can see on the right side that MinION and PacBio are like more likely to be used when we are looking for long reads. And later on in this presentation, I will explain the importance and the difference between short reads and long reads. Here are the cost comparison. Like you can see, like in 2001, you need to like about $100 million to do sequencing for one human genome sample. While in 2017, the cost has been reduced to about like 1,000. And besides the cost is also the time required for this. I mean, in 2001, you will need like few years to get the whole genome sequenced, while in 2017 until nowadays, you can just get the human genome sequencing for the individual, fully sequenced for an individual in like few hours or maximum like days. As we can see here, like the cost of the first human genome project was about 3 billion, while nowadays like a genome will cost about $100. It's a huge reduction in the cost, effort, and time. And now we're talking about different technologies, different capabilities, and need to choose the technology that suits our application. I mean, if I'm doing like kind of experiments on bacteria or on a small model, or the target of my project is to find mutation within the genome of like a simple or prokaryotic thing. So you don't have to spend lots of money. I mean, these equipments with high capabilities that reduce the cost of the human genome sequencing, but it's also like it's inexpensive machines in the first place. So when you have it in a very well established core facility, you can start using it. But sometimes you don't need it. You can just send your sample to this. So not every single institute or department have to have one of these devices. I mean, one central unit in the university or even in the region might be more than enough to cover all the demands from the researchers in the area. And the application that you are after, if all like what are you want to do with the sequencing that you are trying to do? Is it all type of genetic variation, single nucleotide variants, or you are after a gene panel, discover science or diagnostics, germline variation, or also somatic? And these are like will decide based on the request from your research which platform and which technology to use. In addition to that, we need also to finance expertise, bioinformatics capacity, and turnaround time. خلي أوضح هذا السلايد يعني مهم بالعربي. أنا قلت يعني ذكرت هذه السيكونسينج ماشين، وطبعا هذه موجودة صح، هي خفضت الكلفة مع عمل السيكونسينج، لكن بنفس الوقت هي غالية لما تشتريها أول مرة. يعني مثلا نتكلم عن الإلومينا نوفاسيك، يعني سعرها تقريبا £1,000,000. فهي الكلفة مالت السامبل رخيصة، لكن حتى تبدي أن تنشأ يعني يونت مجهزة بهذه الأجهزة هو أيضا الموضوع مو يعني الكلفة مالتها راح تكون نوعا ما عالية. لكن ممكن أنه مثلا على حد معلوماتي مثلا بالعراق يمكن إذا أكو جهاز واحد راح يغطي حاجة كل الجامعات إذا يشتغل عليه تيم كفو ومستقر. كذلك اكو جهاز رخيص جدا وصار يعني عليه نقاش في احد الجروبات قبل فتره، اللي هو المناين يعني سعره 1000 أو 2000 دولار يعني، او يعني حتى لو 5000 دولار ويا الرياجنتس مالته. هو جهاز جدا رخيص، وايضا يعني متطلبات عمله يعني قليله، يعني كمبيوتر وبعض المواصفات الخاصه بالكمبيوتر. لكن مشكلته انه كميه الديتا اللي يولدها هذا الجهاز كميه هائله ونسبه خطا عاليه، فتحتاج الى بايو انفورماتكس تيم يعني كلش قوي حتى يقدر يساعدك في يعني تنظيف النتائج وجعلها ممكنه بالتطبيق. لذلك يعني من تريد تستخدم او من تريد تشتغل شغل معين لازم تختار البلاتفورم المناسب، لازم ايضا تراعي الكلفه والخبرات المتوفره عندك. هنا طبعا لقطة سريعة عن عملية النكست جنريشن سيكونسينج. نبدي بالسيكونسينج اللي بعدين يعني راح تشوفوا انه يجمع لنا الداتا اللي هي عبارة عن ايمج بروسيسنج، بعدين راح يولد هذه الريدز، بعدين هذه الريدز السيكونسز اللي حصلناها راح نسوي لها ريد الاينمنت اند ذن، وبعد الالاينمنت مالتها يعني الالاينمنت يعني مقارنتها بالريفرنس جينوم، وبعد ان قارناها بالريفرنس جينوم نبدي نستكشف الفاريانت، نسوي فاريانت ديتكشن، وبعدين هذه الفاريانتس اللي راح نحصلها ايضا نعمل لها برايورتايزيشن حتى نشوف اي من هذه الفاريانتس هو حقيقي وممكن ان يستخدم. النكست جنريشن سيكونسينج بيزكس يعني ايضا اذا مرينا بخطوات سريعه لازم عندنا الجينوميك دي ان اي، نبدي نسوي له فراجمنتيشن، بعدين بعمليه كيميائيه انزيميه نلصق له الادابتر لايجيشن، بعدين نخليه على الفلو سيل الخاصه بالجهاز، وهاي الفلو سيل راح تبدي يشتغل عليها سيكونسينج باي سينثيسيس الطريقه تسمى. هذا المثال هنا هو عن اليومينا شورت ريد سيكونسينج، واعتمادا على الفلورسنت داي اللي موجوده ضمن الفلو سيل واللي راح ترتبط ويا الفراجمنتس اللي يتم بنائها، راح يصير عمليه تصوير او كابتشرنج للفلورسنت coming from each sequencing or synthesis event. اكو كاميرات راح تلتقط السيجنال، وبعدين هذه السيجنال تتحول يعني يصير لها بروسيسنج وتتحول الى ترانسليت تو يعني NGS reads اللي هي قواعد النيتروجينية الأربعة حسب نوع الفلورسنت. بعدين الخطوة اللي بعدها احنا حصلنا يعني كمية هائلة من السيكونسينج فراجمنتس، لازم احنا هذه السيكونسينج فراجمنتس نسوي لها مابينج للهيومن جينوم او الريفرنس جينوم حتى نقدر يعني نعرف انه هذه القطع هي مكاناتها الحقيقيه وين ضمن الجينوم، لانه بعدين نحن من نريد نجي نسوي الاناليسيس لازم نعرف انه هذا الفاريانس اللي حصلناه وين واقع ضمن يا كروموسوم ضمن يا جين، وين الموقع مالته بالضبط، وهذه كلها طبعا المسائل يحلها البايو انفورماتكس تيم. And there's also another thing called like de novo assembly. احنا نبدي بنسوي هذه الاسمبلي لهذه الفراجمنتس اللي حصلناها حتى نقدر نسوي calling للفاريانس ونرتبها بالاوردر اللي مطلوب حتى يعني نقدر نسوي لها مقارنة مع الريفرنس جينوم. طبعا العملية هذه اسهل لما نكون يعني بدنا نستخدم واحده من تقنيات اللونج ريدز، يعني مثل البازلز الصغيره. اذا انت البازلز مثلا 4 قطع وتريد تركبها، لو اذا مثلا 400 قطعه نفس المبدا، ترتبها وترجعها وتقارنها مع الجينوم. وهنا عندنا مثال شلون راح نسوي الالاينمنت او الالايننج لهذه السيكونس اللي حصلناها مع الريفرنس جينوم، وراح نشوف انه اكو تباين باعداد الفراجمنتس او القطع، يعني من حيث الطول ومن حيث العدد لكل قاعده يعني تم تغطيتها ضمن السيكونسينج بالجينوم، وهذا نسميه الدبث او العدد مرات السيكونسينج لكل فراجمنت. This is what we call individual sequence reads, and and coverage can be determined per nucleotide position، يعني حسب كل النيوكليوتايد كم مرة صار لها sequencing. وأكو طبعا هناك gaps اللي هي المناطق اللي ما صار sequencing ضمنها، وهذه الحالة تكون موجودة أكثر شيء بالهول إكزوم سيكونسينج، لأنه إحنا نركز بالهول إكزوم سيكونسينج على الجزء المشفر من الجين فقط وليس الأجزاء الأخرى. حتى نيجي نسوي الفاريانت حتى نسوي الفاريانت ديتكشن ثيوري، يعني عندنا مثلا هذا السيكونس، لما نقارن هاي السيكونس اللي حصلناها مع الوايلد تايب جينوم او الوايلد تايب سيكونس، راح نحصل الصوره اللي هنا على اليسار اللي هو عندنا 3C و3G، فمعناها احنا عندنا هيتروزايغوس فاريانت، بينما الصوره اللي على اليمين حصلناهم كلهم T والاصلي هو A، فمعناها اكو هوموزايغوس فاريانت هنا موجود. طبعا هنا هذا الريفرنس جينوم اذا حصلنا يعني عندنا 3 ريدز هنا، اثنين من عندها مشابهه للريفرنس، واحد مختلف، يعني هنا عندنا راح يكون نوع من الضبابيه، اكو واحد فاريانت تو ريفرنس ريد صح النسبه مالته البيرسنت 33%، بس لو كفرج هنا والريزلتس راح تكون نوت ريلايبل، فما نقدر نقول انه هذا الفاريانت موجود لانه عدد الريدز قليله وما ندري اذا يعني اذا هو حقيقي او لا. لما يكون عدد الريدز للقاعده الواحده اللي احنا يعني صار بها او صار لها كول از فاريانت يعني اكبر، مثلا هنا راح نشوف انه اكو 4 فاريانت و4 ريفرنس، فمعناها ممكن انه نقول هذا يعني صح لو كفرج بعده، بس ممكن انه يكون هيتروزايغوس فاريانت لانه اكو تكافؤ بالعدد بالحالتين. لذلك احنا يعني بالسيكونسينج باستخدام الاليومينا نسوي البيرد اند سيكونسينج حتى يصير سيكونسينج لنفس القطعه من جهه اليسار ومن جهه اليمين، لانه احنا ايضا صح هي يعني تقنيات السيكونسينج الحديثه هي تقنيات قويه ووفرت يعني كميه هائله من المعلومات، لكن احنا ايضا ما ننسى انه معظم هاي التقنيات تعتمد على البي سي ار، والبي سي ار اكو باحتماليه الخطا او المس انكوربريشن تكون يعني وارده وموجوده، لذلك يعني لازم نكون نسوي دبل تشيك ونتاكد من كل شيء. الفاريانت بالجينومز ايضا لها اهميه كبيره، خصوصا يعني القطع الكبيره هذه لازم يعني نستخدم التقنيات المناسبه حتى نقدر نكشف عن الكوبي نمبر فاريشن، وايضا هذا يعني النمبر اوف كفرج والكواليتي اوف سيكونسينج اللي راح نحصلها حتى نقدر نكون يعني على ثقه بهذه النتائج. single molecule without amplification هذه أعتقد راح يعني تكون واحدة من التقنيات اللي لها يعني استخدام كبير في الأيام القادمة والسنوات القادمة، لأن هذه التقنية نقدر أنه نسوي جينوم سيكونسينج بدون إجراء أي تحضيرات أو أي تفاعل PCR، لذلك هي راح تقرأ لنا السيكونس بناء على ما موجود فعلا من الدي ان اي اللي عزلناه مباشره، نسوي له سيكونسينج بدون اي عمليات او يعني تضاعف بالبي سي ار، وهذا راح يقلل نسبه الاخطاء. طبعا ايضا البروميس اوف single molecule sequencing amplification PCR sequence include artifacts. DNA needs to be chopped in small fragments. It takes time to be, and it is expensive. Sequencing of one molecular chromosome at a time is potentially ideal, especially analyzing complex genomes, and major advantage long-read sequencing, major challenge raw sequencing accuracy, and major combined باكبايونكس فورتانوبور. هذه الشركات الرائدة في مجال اللونج ريد. سوري أنا راح أضطر يعني شوية أمشي بسرعة لأنه الوقت داهمنا. الادفانتجز اوف ذا لونج ريد سيكونسينج انه تساعدنا في فهم الستراكشرال فاريشن، الريبيت اكسبانشن، الفيزنج والجينز. اكو يعني بعض التفاصيل لما نستخدم الشورت ريدز اللي هو هنا ما راح ممكن انه نلقى يعني تحليل واضح أو تحليل سهل ومباشر للفاريانس اللي موجودة ضمن هذا الجين معين. لكن لما نسوي سيكونسنج للجين في كقطعة واحدة أو قطعتين مثل ما نشوف هنا في حالة اللونج ريز، راح نقدر ننطي استنتاج أكبر، أو راح نقدر نسوي الفيزنج. أي الفيزنج يعني إحنا نستخدمه في حالة الدينوفا ميوتيشن، يعني الطفرات اللي تحدث في الفرد واللي هي غير يعني مو موروثة عن طريق الأب والأم. والفيزنج راح يساعدنا أنه نعرف الأليل اللي صارت به هذه الطفرة أو الفاريشن، هل هو مصدره من الأب أو من الأم. دراسات يعني لربط هذه الأمور مع أيضا في كشف السيدوجينز اللي هي شبيهة للجينات واللي توهمنا أنه فعلا يعني أكو فاريشن يعني موجود أو فاريانت، بس هي بالحقيقة سيدوجين. الأسئلة اللي نسألها which NGS can be used now? Can we sequence all known disease genes of genetically heterogeneous disease in parallel? Hereditary breast cancer, hereditary blindness, and so can we sequence entire candidate disease gene loci from linkage studies, homozygous mapping? And can we sequence the whole exome, all exome of gene? طبعا هنا الأسئلة هذه يعني اللي تطرحها أنه أنا شنو أستخدم يا تقنية حتى أقدر أحل المشكلة. إذا أنت يعني إذا مثلا هسه حاليا في الكانسر بدراسات الكانسر اكو جين بانل اللي هي مجموعة الجينات اللي من خلال الدراسات السابقة معروفة انها تأثر او لها دور في عملية الكارسينوجينيسيس، فجمعوها كلها في بانل واحد، وانت بدل ما تروح يعني تبحث بالجينوم كله راح تركز فقط على هذا الجين. او احنا مثلا حتى ضمن الجروب اللي احنا نشتغل عليها يعني جمعنا الجينات اللي مسؤولة عن الميل انفيرتيليتي كلها في بانل واحد، وسوينا يعني بانل من 132 جين وبلشنا دراسات مركزة عليها. وطبعا كل هذه التقنيات أو الطرق يعني هي ممهدة. إذا ما عندك يعني قاعدة بيانات ثابتة للجينات اللي داخلة بالمرض اللي راح تدرسه، فممكن تروح إنه على باتجاه الهول إكزوم سيكونسينج. ومن نقول هول إكزوم سيكونسينج يعني معناها تدرس كل الإكزونات في كل الجينات، واللي تمثل تقريبا 1% of the genome. هذه الطرق المختلفة اللي احنا نستخدمها: fractionation of whole genome library to isolate exome fragments، أو molecular inversion probe اللي زميلي دكتور ريتشارد will explain، PCR amplicon اللي ممكن نقدر نستخدمه مباشرة. وهذه كل واحدة من هذه الطرق طبعا لها advantages and disadvantages. ممكن انه احنا نسوي الجين بانل مثل ما ذكرنا ونركز في دراستنا على شنو الجينات اللي احنا عندنا انترست بيها وشنو هي الطريقه المناسبه. الهول اكزوم هو الهول اكزونز للجينوم اللي هي 1% من الجين، والكودينج سيكونس اوف هيومن جينوم تقريبا 200,000 exon can be sequenced and analyzed in one experiment. هذه طبعا للشخص الواحد هي 200,000 إكزون، لكن أنا قلت في بداية حديثي أنه ممكن أنه نسوي whole exome sequencing for 96 people at the same experiment and in the same tube. والطريقة طبعا بها تبدي بعملية عزل الجينوم بنوعية بكواليتي وانتجريتي جيدة، بعدين نسوي له fragmentation، بعدين نسوي له hybridization, washing, pull down, and then capture the DNA and then run it on the DNA sequencing platform. And then after we get the sequence, we do the mapping, alignment, and variant calls and filtration and these things. And then we can do the mapping and annotation to exome sequence reads, and these will be aligned with the and mapped and aligned with the reference genome. And we can see the coverage here of the different exons parts. And when there is a variant that we need to call, we will see it in here, down here. ايضا التقنيات اللي دا توصلنا صح هي تقنيات يعني بها kind of magic وانه دا تساعدنا في الوصول الى حلول ونتائج جيده في عمليه كشف الاسرار الموجوده بالجينوم، لكن التطبيق مالتها او الوصول الى النتائج المقبوله هو مو straight forward. You need to do some optimization all the way from the DNA extraction to the variant calling. واحدة من الأشياء اللي هي طريقة الانريتشمنت، اللي هي الخطوات اللي نستخدم بها تقنية الPCR حتى نحضر الجينوم لعملية السيكونسينج. عادة تصير نوع من الدوبليكيشن أو البايس أو something like that. So we need to optimize our enrichment method to ensure that all the samples has been treated the same way, they are the same quality, and they generate like the same good quality of the library, so we can rely on it. You don't want to see a huge difference between samples, that one of the samples covered 100x times and the other one just 10 or something. You need to get them all very, very close and homogeneous. And the variant notation is very like, it's not a very complicated, but there are too many details included and too many databases that you can use in order to reach to the conclusion that the variant you are calling is the right one, and you can rely on this and validate it, and you can then report it as the cause of the disease. And that's about 22,000 coding variants are identified in each individual. That differs from the wild type sequence. And when we call the variant a human genome mutation database, and PubMed, we search the mouse knockout phenotype. We can do other exome conservation scores, online Mendelian inheritance models, and too many steps that we need to use, whether we are looking for variant-based annotation or gene-based annotation. And we do all these different sort of searches and annotation for the variants before call it, or before, I mean, report it and confirm it that this is as the cause of the disease. The development in next generation sequencing, the quality, short read sequencing reliable for most application, average sequencing coverage reliable for detection of pointing mutations, long read sequencing better for structural variation, repeat expansion, but high error rate for single nucleotide variation detection still exists, especially in the long reads. For the throughput and the speed, short reads technology allow exome and genome sequencing in days, and thousands of genome can be sequenced in individual system annually. Long reads genome still take more time and have less throughput. While the cost is short read technology or 100x coverage exome, it's about €300 these days. 30x genome is about like 1,200 euro. Long read genome 30x, and it depends on the coverage that you are after, but the cost kind of reasonable these days, but we are expecting to go down more and more because of the competition between these different companies. And because when we do genome sequencing, all variation in one experiment, that's what we can get, and the importance of the accuracy and completeness, all genome, all variation, speed and price, and that's what the new technologies can offer. And you can start extracting DNA from variable, like very different samples, like from blood or saliva or whatever. Why you perform whole genome sequencing? If you consider genetics may play a role in your patient, why not read the entire book? Why settle for studying what we now know, and we still live in the dark ages of genetics? There are too many stuff. يعني احنا يعني حاليا اكو توجه باستخدام تقنيه الهول جينوم سيكونسينج. الهول جينوم سيكونسينج تكشف لك كل شيء، يعني اذا انت يعني المشكله اللي تبحث عنها ماكو وجود لها بالليترشر، ما تقدر تسوي لها يعني تختصرها او تركز على جزء معين من الجينوم، الهول اكزوم سيكونسينج ما حلك المشكله. فالافضل انه تروح باتجاه التقنيه الشامله او تقرا read the whole book the entire book. and مع ذلك لا يزال الموضوع يعني حاليا يعني بدايته احنا يعني قلنا انه الكوست نزلت من 300 بليون الى 100 دولار، بس ستيل طبعا اكو يعني بعض الصعوبات، بعض الامور لا تزال يعني مو مثاليه مو 100%. مع ذلك اكو احنا يعني لما ندرس الجينات والجينوم والاكزونز we're still looking at 2% of the entire genome, so there's still a lot to do with the remaining 98% of the genome. على مستوى العالم, there are like genome sequencing centers established around the world. Even the very, very well-developed countries now starting focusing more on these technologies. We can see like in 2015, President Barack Obama announced, launched a new precision medicine initiative to bring us closer to curing disease like cancer and diabetes. And the Genomics England also started in 2017 to sequence 100,000 genome, and this job already been done. Now that we have the database for 100,000 whole genome sequencing from UK patients and parents, and we'll try to finish my talk. What you are telling the participant: information about patient main condition, information about additional serious and actionable condition, carrier status for non-affected parents of children with rare disease. طبعا اكو هنا صارت اتفاقيه مع الاشخاص اللي تم اخذ النماذج منهم على شنو الاشياء اللي نخبرهم بها نتائجها، او شنو الاشياء اللي ماكو داعي انه نحللها او نروح خلالها. We are like increasing move towards exome and genome sequencing, further use of patient and trial data. طبعا حاليا يعني صار تركيز بالعالم على استخدام التريو، يعني التريو موديل بالدراسات خصوصا في الجينومكس يكون يعني افضل، لانه انت تسوي سيكونسينج للشخص وتسوي ايضا الاب والام لمعرفه انه يعني هل المرض اللي يعني بالنسبه للامراض الوراثيه اصبحت معروفه، فما ماكو داعي ترجع للبيرنت. لكن اكو حق نسميه الدينوفو ميوتيشن، والدينوفو ميوتيشن ما راح نقدر نعرف انه الميوتيشن اللي حصلناها is it de novo or not unless we have complete sequencing data from the parents. And here I want to thank also my team, the Professor Joris Veltman and my colleagues in the Institute of Genetic Medicine, and now currently it's called Institute of Bioscience. And thank you for the audience for your listening and for your patience, and I hope I delivered my message through to you. And apology for Dr. Richard, as I invaded some of, I took already still some of his time. So thank you very much.
الو عفوا دكتور، راح الانترنت من عندي. شكرا جزيلا دكتور. Thank you ever so much. I do. Can you hear me, doctor?
Yeah, I can hear you.
I do agree. Actually, we're in the dark age of genetics. It's really, really insightful. Thank you ever so much for your participation.
Thank you.
Thank you. Now, I would like to welcome the next keynote speaker, Dr. Richard Gallen. It's a research associate, a cancer prevention research group Institute of Genetics, Newcastle University. Dr. Gallen, are you with us?
Yes, good morning.
Good morning, sir. How are you?
Very well, thank you, and thank you for the kind introduction.
No problem.
I will share my screen
Go on.
if I can. Okay, hopefully you can see my screen now.
Yes, we can. Yeah.
Fantastic, thank you.
Okay.
So, my name is Richard Gallen, and as I've already been introduced, I'm a research associate in the Cancer Prevention Research Group at Newcastle University. And today I'm going to be talking to you about the application of next generation sequencing to high throughput microsatellite instability testing. First of all, I'm going to give you a little bit of background on our research group. So the research group is led by Professor Sir John Byrne, Dr. Mauro Santibanez-Careth, and Dr. Mike Jackson. And one of our main interests is in the prevention of cancer, and from that stems several other research interests. So this is one of our most recent publications where the CAP2 clinical trial, which was started back in 1998, has now reached its 20-year follow-up period, meaning that we have 20 years worth of data on the participants in that trial. And the participants in that trial were given aspirin or placebo to see if there was any effect of aspirin on the colorectal cancer risk in this particular patient group. And what the CAP2 trial showed was that patients taking 600 milligrams of aspirin per day do have a significant reduction in their risk for colorectal cancer. Now, aspirin does have side effects, so it's very important that you can identify at-risk groups, i.e. patients who have a very high risk for colorectal cancer, so that the benefits that they get from the reduction in their cancer risk outweigh the risks of the daily aspirin intake. So this has been one of the focuses of our research group for a long time, and long before I joined the group. But from this comes our interest in identifying patients to whom we can give aspirin, which will be one of the main subjects of our talk. In 2015, I started my PhD in the research group, and this is just showing everybody who was a member of the group at that time, as well as a couple of our collaborators. So just as a quick overview of my talk today, first of all, I will go over how and why we test for microsatellite instability in cancers, particularly colorectal cancer. And then I will present a tumor sequencing-based microsatellite instability assay, which can use as few as 6 microsatellite markers, which therefore allows for very high throughput diagnostics. I do have a couple of disclosures to make. Newcastle University owns 2 patents that cover the microsatellite markers that are used in the assay that I will present to you, and I'm named as an inventor on one of those patents. So mismatch repair deficiency is a phenotype that we find in approximately 16 to 17% of all colorectal cancers. It's typically found in colorectal cancers that are found in the right side of the gut or the proximal side of the colon. These cancers also show an increased immune cell infiltrate, and probably due to this increased immunogenicity, they have a better prognosis relative to cancers that do not have this phenotype. In addition, the strong immune response against these cancers means that they respond very well to immune checkpoint blockade therapy, which is a relatively new therapy that has really become a point of interest since about 2015, when the first paper was published showing that mismatch repair deficient colorectal cancers were highly responsive to pembrolizumab, which is also marketed under the name Keytruda. Mismatch repair deficiency in colorectal cancer is also associated with a hereditary cancer predisposition called Lynch syndrome. Lynch syndrome is caused by pathogenic variants in the germline of the patient, which affect one of the 4 mismatch repair genes, MLH1, MSH2, MSH6, or PMS2. These patients have an exceptionally high risk for colorectal cancer. Up to the age of 75 years, they have something like a 50% chance of getting colorectal cancer. In female Lynch syndrome gene carriers, they also have roughly a 50% chance of getting endometrial cancer up to the age of 75 years. They also have high risk for a number of other cancers, urothelial, gastric, small bowel. There is suggestions that both breast cancer and prostate cancer might have slight increases in risk in this patient group as well. Focusing on colorectal cancer, Lynch syndrome accounts for approximately 3% of all colorectal cancers. And what we see in the tumours of these Lynch syndrome patients is that the second healthy copy of their mismatch repair gene, which is affected in their germline, that second healthy copy gets mutated in the tumour, and therefore they lose one of the mismatch repair proteins from being expressed due to 2 mutations in both alleles of that gene. This then creates this state of mismatch repair deficiency in the tumour. In the sporadic setting, i.e. in the general population where there isn't a hereditary risk for colorectal cancer, what we find is that it's typically the MLH1 gene and it gets methylated, which silences it, and that leads to mismatch repair deficiency. So mismatch repair deficiency occurs in the context of Lynch syndrome, but also sporadically in the general population. And therefore Lynch syndrome accounts for approximately 20 to 25% of all mismatch repair deficient colorectal cancers. High levels of microsatellite instability, or MSI high, and tumour mutational burdens that exceed 10 mutations per megabase are characteristic of mismatch repair deficiency. So microsatellites have a variety of different structures, and here I'm showing mononucleotide repeats, dinucleotide repeats, etc. And a brief description of what a microsatellite would be a short sequence of repetitive DNA. The reason why these mutate at a very high rate when you have mismatch repair deficiency is because the mismatch repair system is integral to the correct replication of these repetitive regions of DNA. So taking this mononucleotide repeat as an example, during DNA replication, the polymerase is synthesizing the daughter strand using a template strand. And what can happen at these repetitive sequences is that you get a slippage of the polymerase. The polymerase dissociates from the DNA, and what happens is the 2 strands of DNA re-anneal together, and that structure, even though normally that would be very unstable, in the case of repetitive sequences of DNA like this, that microsatellite structure is stabilized despite the deletion loop because of the annealing of the repetitive sequences. And if replication continues, we now have, in this case, 2 nucleotide deletion within that microsatellite. So in the case of mismatch repair deficiency, that mutation would not be detected. However, where the mismatch repair system is functioning, the mismatch repair proteins detect that lesion by binding to it. They then excise the daughter strand, which then allows the polymerase to have another go at repeating the replication of that microsatellite. And that mismatch repair system will continue to work until replication is correct. So when you have mismatch repair deficiency, your mutation rate in these repetitive sequences increases by over 1,000-fold, and therefore high levels of microsatellite instability is a well-established biomarker of mismatch repair deficiency. So there are several ways in which we can test for mismatch repair deficiency. One way is to use immunohistochemistry. When you have mutations in the mismatch repair genes, very often the protein is therefore not expressed. And therefore, if you try and stain for the protein, if you have mismatch repair deficiency, you will see an absence of staining. So in the image here, this is an adenoma that has mismatch repair deficiency. You can see the brown stain, which is representing mismatch repair proteins being expressed within the intramucosal tissues within the mucosal epithelium of this adenoma. And where you see the pale blue, that is the adenoma epithelial cells, which are lacking mismatch repair protein staining. You can also use microsatellite instability testing. The most common technique used is PCR fragment length analysis. So you use polymerase chain reaction to amplify a panel, in this case of 5 microsatellites. These are all mononucleotide repeats. And then you run those on a capillary electrophoresis machine. they are fluorescently tagged. So then you can look at the traces of these, of the fragments as they flow through the capillary electrophoresis machine. What you can see here is you get these stutter bands, and these stutter bands occur because microsatellites in a PCR reaction are very, very mutable. And so each one of those stutter peaks can represent an error in the PCR reaction, introducing insertion or deletion mutations. However, in some tumors, you can see that you get these extended shoulder peaks, as we can see in the figure here, highlighted by the 2 red arrows. And in this case, that is an indicator that the tumor has high levels of microsatellite instability. You can also use next generation sequencing to detect microsatellite instability. And you use short read sequencing typically, and you capture your microsatellite, you sequence them, and you look at the distribution of microsatellite lengths detected within your reads. And that can be converted into a bar chart, such as the one on the right here, where you can see that in the microsatellite stable tumor or mismatch repair proficient tumor, more than 90% of the reads are of the 0 microsatellite length, meaning 0 length change relative to the reference sequence. Whereas when we look at the microsatellite instability high colorectal cancer, we can see that there are many other reads of different lengths of microsatellite, typically deletions. And that is therefore an indicator that that microsatellite is mutated. So we have 2 main needs for mismatch repair deficiency testing clinically. The first of these is to try and identify patients with Lynch syndrome. Identifying these patients is very important because they, due to their very high colorectal cancer risk, can benefit from, for example, daily aspirin intake, as I showed at the start of the presentation. They can also have regular colonoscopy, and regular colonoscopy every 1 to 2 years can identify precancerous lesions, and then polypectomy can be used to remove those lesions before they develop into a cancer. You can also have risk-reducing surgery. So for example, a patient who has already had a colorectal cancer, rather than having just a segmental colorectal resection, they may have a full total colectomy, i.e. the full colon removed, which therefore reduces their risk of metachronous colorectal cancer. So there are several guidelines in the US, Europe, and the UK, as well as other countries that state that you can start to screen for Lynch syndrome by mismatch repair deficiency testing of all colorectal cancers. If those cancers are mismatch repair deficient or microsatellite instability high, you can then do a number of other tests, such as MLH1 promoter methylation testing or BRAF V600E mutation testing, to identify the sporadic or the mismatch repair deficient tumors that are from the general population and remove them from the screening pipeline. If those cancers are unmethylated and lack the V600E mutation, there's a higher likelihood that those are going to be due to Lynch syndrome. So then you can do germline mismatch repair gene testing to confirm a diagnosis. The second key need is that immune checkpoint blockade therapy has shown to be very effective in mismatch repair deficient cancers, and therefore being able to identify them allows you to give the patients the correct therapy. So at the moment, if we compare the 3 different methods, in general, immunohistochemistry and microsatellite instability analysis by fragment length analysis are both relatively cheap, both highly sensitive and specific for detecting mismatch repair deficiency. However, they're relatively low throughput, and this is partly because they are not automatable. It requires an expert to interpret the results. You need, for example, a pathologist to interpret the results of immunohistochemistry, and you would also need a trained clinical geneticist to interpret microsatellite instability traces by fragment length analysis and capillary electrophoresis. The other big disadvantage of immunohistochemistry and fragment length analysis is that they cannot test other markers in one go. Therefore, for example, you could not test the BRAF gene or MLH1 promoter methylation. And therefore, the Lynch syndrome screening pipeline is a multi-step process. With microsatellite instability testing by next generation sequencing, you get equivalent sensitivities and specificities. However, the cost is much higher. The protocols are much higher throughput. As Bilal has shown to you, you can sequence potentially hundreds or thousands of different samples on one sequencing machine. The analysis can be automated, therefore removing the need for experts to interpret the data. You can also test other markers. So for example, as well as testing microsatellite instability, you can already be sequencing the mismatch repair genes to try and identify any variants in the tumor, which you can then later compare to when you sequence the mismatch repair genes in the germline. And therefore, you can reduce your Lynch syndrome screening to effectively one step. So looking at the statistics of how well mismatch repair deficiency testing has been taken up by clinics, in the US, only 43% of young colorectal cancer patients who are at most risk for hereditary disease such as Lynch syndrome were tested for mismatch repair deficiency. And a questionnaire of 509 gastroenterologists found that the main two reasons for this lack of uptake of testing was the cost of testing and the unfamiliarity with interpreting the results. Therefore, these are clearly 2 points that have to be addressed. And what we aim to do in the project that I will present to you now is develop a next generation sequencing based microsatellite instability test that has all of the advantages of next generation sequencing, but a low cost. And therefore it can be applied to the clinic broadly. So if you want to develop any diagnostic test, you have to consider several key points. And this is in the context of microsatellite instability testing, but some of these things will apply to other applications as well. So for example, you have to consider what sequencing platform you wish to use. And I think Bilal gave a much more thorough and better explanation of this than I will be able to now. But for us, looking at microsatellites, just as an example, iron torrent technology wouldn't be great because they are known to have a higher error rate at microsatellites. It's also very important to pick the correct markers. So all microsatellites, depending on their structure and their genomic context, behave very differently to mismatch repair deficiency. Some are more or less sensitive for that. You also have other things to consider, such as polymorphisms in the microsatellites. If they are highly polymorphic, then you will need matched normal tissue to be able to interpret your microsatellite instability results accurately. Whereas if they are monomorphic, i.e. there are no normal variations in the germline for those microsatellites, you can call mutation in them with higher accuracy. You also have to think about your classification method. So most methods use a system where they look at each microsatellite, say whether or not that microsatellite is unstable, and then work out what proportion of all the microsatellites that they've looked at have instability or have mutations. And therefore, if you have over a certain threshold, typically people use 30%. So if you have more than 30% of your microsatellites showing instability, that tumor is therefore microsatellite instability high and therefore mismatch repair deficient. You also have to consider how many markers you need. So by next generation sequencing, you get lots of errors introduced into the microsatellites. And typically you need to have lots and lots of markers. But if you have lots and lots of markers, that means you need relatively large panels, which therefore means you are sequencing more per sample, which therefore means the per sample cost increases. So there were two PhD students in the group before me, Lisa Redford and Ghanem Al-Halal, and they developed a novel approach to microsatellite instability classification of tumors from next generation sequencing. First of all, they selected markers. They used mononucleotide repeats because these have the greatest sensitivity and specificity, and they can also detect MSH6 deficiency, whereas other structures of microsatellites such as dinucleotide repeats, trinucleotide repeats, the MSH6 protein is not involved in the repair of those, and therefore they are insensitive for MSH6 deficiency, whereas mononucleotide repeats can detect that. We also chose relatively short markers. These were chosen because they have reduced sequencing and PCR error, which makes it much easier to interpret those markers to see if they have instability or not. We also chose markers that had a common single nucleotide polymorphism within 30 base pairs, which would then allow us to map a read not only by the length of microsatellite, but also to which allele within the patient's germline that microsatellite deletion or insertion was associated with. And I'll come back to why that's useful shortly. We also wanted monomorphic markers so that we didn't need to look at matched normal DNA. So Lisa and Ghanem identified 120 different markers. They then went through a process of identifying the best of these markers, which I will skip over quickly. Only point I will highlight here is that we use a relatively high read depth. So previously Bilal was talking about read depths of maybe 30x, maybe 100x, whereas here we're using relatively high read depths of 5000x, and that is because microsatellites have such a high error rate of sequencing and also PCR that you need to have a high read depth to be able to distinguish the signal of a true mutation from the noise of error. So using a relatively small cohort of 60 colorectal cancers, Lisa and Ghanem's work identified 17 markers that were the superior set based on their receiver operated characteristic area under curves. They then had a classifier training cohort where they had 139 colorectal cancers of known microsatellite instability status. And then they use both the microsatellite deletion frequency and the allelic distribution of deletions to classify these samples. So if we look to the figure on the right, the bar chart, this is colored according to the SNP allele that was neighboring that microsatellite, with then the microsatellite's length distribution shown before and the proportion of reads that have those different lengths on the y-axis. What we can see here is in this mismatch repair proficient tumor, these shoulder peaks, as you would see in the capillary electrophoresis for fragment length analysis, representing error. And what we can see here as well is that by the color of these bars, you have a roughly even distribution to the C allele of the SNP and the T allele of the SNP. And therefore, even though we have that deletion, the equal distribution between the two alleles suggests that it isn't actually a true deletion and it's just sequencing error, because error will occur equally across both alleles, whereas deletions that have occurred biologically in the actual tumor will track with one allele or the other. And therefore, when we look at this mismatch repair deficient colorectal cancer, we can see two things. One, we can see that the frequency of deletions has increased, which is an indicator of microsatellite instability, but also that those deletions are specifically tracking with the C allele. So we can therefore use the SNP to improve our confidence that what we are observing is true microsatellite instability rather than a PCR or sequencing artifact. I'll rush through these next few points. This is basically a detail of how we do the classification, but the long and short of this classification is that it's a naive Bayesian approach where we actually use the probabilities at an individual marker level to determine whether or not we are observing instability in that marker. And because we are doing this, this means that we're treating all of the markers differently and accounting for their different behaviors in the context of mismatch repair deficiency, which increases the sensitivity and specificity of the method. What we then do is, having defined a probability of the tumor being either mismatch repair deficient or proficient for each marker, we take the product of all of those probabilities, we correct for the prior probability and convert that into a simple score. So using that simple score, we say that if we have a score above 0, that means that the tumor has microsatellite instability or has high levels of microsatellite instability. If the score is below 0, we say that it's microsatellite stable. And Ghanem and Lisa's work showed that in the training cohort, which was used to define all of these probabilities, reclassification of those same samples using the classifier was 100% sensitive and 100% specific. In the validation cohort, they achieved 97% sensitivity and 97% specificity. And afterwards, we resolved the discordance of our result with the reference assay in 3 of those 4 samples. So the sensitivity and specificity was actually higher than that. However, there were limitations to the assay developed by Ghanem and Lisa. They were using a singleplex PCR, which means that it was very low throughput and very high cost because they were individually amplifying those 17 markers for each sample. And then there was no evidence that the assay was suitable for clinical practice. So at the start of my PhD in 2015, when I took over from Ghanem and Lisa as they finished their PhDs, the aims of my projects were to multiplex the method that they were using, therefore to make it high throughput and reduce its cost, and then show that the assay was suitable for clinical deployment. So we selected a total of 24 mononucleotide repeats and the BRAF locus. The reason why we selected BRAF is that it can be used in Lynch syndrome screening, and therefore by including that into this assay, we can make this a one-step Lynch syndrome screening pipeline rather than having to do microsatellite instability and BRAF testing separately. So to do that multiplexing, we used single molecule molecular inversion probes, which Bilal touched on earlier, but I'll go into a little bit more detail here. So the basic structure of a single molecule molecular inversion probe is like two PCR primers, except that they are joined by a backbone. There's also this MB, which stands for molecular barcode. And basically, this is a unique molecular tag of random nucleotides, which is unique to each molecular inversion probe molecule. You then use these molecular inversion probe molecules in a four-step process. In step one, the probes anneal to your single-stranded template DNA. And as you can see, the two targeting arms anneal to the same strand of DNA, unlike in PCR, where your PCR primers anneal to separate strands. In the second step, you add in a DNA polymerase and a DNA ligase to fill in the gap and seal the nick. So you now have a circularized probe. By exonuclease treatment, you can get rid of all of the linear DNAs, including the excess template DNA and any unligated molecular inversion probes. And then in step 4, you do a universal amplification. And this amplification step adds on your sequencing adapters, sample indexes, etc. All the additional sequence that is needed for these samples to be sequenced on an Illumina platform. So when we were developing this assay, we wanted to follow recommendations for the validation of it within the clinical setting. So to validate diagnostic accuracy, we have to have careful consideration of the sample number and type, i.e. they have to represent true clinical samples. So we worked in collaboration with the local NHS services in Newcastle to acquire those samples. You need to have independent cohorts, particularly where you're training a classifier. You need to have an independent set of samples to show that that classifier really does work. We also need to look at reproducibility. You also have to have quality controls built into your assay so that you can detect whether or not a result is reliable or unreliable. And then finally, you need to show additional clinical utility. So for example, in our assay, we use the single nucleotide polymorphisms, not only to help us classify the microsatellite mutations as either true mutations or errors, we can also use those single nucleotide polymorphisms to identify the samples. So if you're having to do repeat testing, you can show that the sample has correctly been repeated. So when we did our classifier training, we used all 24 mononucleotide repeats, and we separate on the x-axis here the original classification of these colorectal cancer samples. And then on the y-axis, we have our assay score. And again, you can see that scores above 0 indicate high levels of microsatellite instability. Scores below 0 represent the microsatellite stable phenotype. And so what we showed was in the training cohort, when we used the classifier to then reclassify those samples, we got 100% sensitivity and 100% specificity. And we also wanted to see how many markers we could get or reduce the panel to and still get accurate classification. And we found that we had redundancy in our panel and we could use only 6 microsatellites and still get 100% accuracy in classification. And then again, that's important because the fewer markers you have, the lower the cost of sequencing. We then had a separate validation cohort of 99 independent colorectal cancer samples. And again, we showed that with either the 24 or the 6 marker panel, we could get 100% accuracy in classification. We then wanted to see what the detection limits of the samples of the assay was, and we did this by sample mixtures. So we took MSI high cell line DNA and mixed that with a control DNA that was extracted from human blood. And by mixing those together to create different percentages of MSI high DNA within the mixture, we could then roughly determine that with the 24 marker panel, we can detect as little as 3% MSI high DNA within the mixture. And with the 6 marker panel, about 6% MSI high DNA in the mixture. And that's very useful to know because typically in a clinical setting, you have tumors that you are testing where there is a mixture of tumor cells, but also there will be normal cells in there. For example, cells from the blood system, such as white blood cells, red blood cells, the endothelial cells of blood vessels. You also have fibroblasts, etc. And so what we can show here is that we can detect very low tumor cell content. We then did some in silico experiments, which I won't go into in detail, but those in silico experiments basically confirmed that this assay is very sensitive to very low tumor cell content. We also wanted to see what effect template quality, i.e. the quality of the DNA sample, and also the quantity of the DNA sample had on classification. So we did some work in the wet lab where we looked at 9 different samples and we diluted those samples down to very low concentrations and then classified them by sequencing those samples using our molecular inversion probe assay, classified them to see what effect the reduction in template quantity had on the accuracy of classification. And this allowed us to determine that roughly a minimum of 75 unique template molecules must be sequenced for classification to be accurate. We can detect or estimate the number of unique template molecules that were sequenced by those molecular barcodes that I highlighted earlier. Again, each molecular barcode is a unique sequence and therefore represents one single molecular capture event. So even if you have 5,000 reads, you can, within those 5,000 reads, you may only have 100 or 200 different molecular barcodes represented. Again, we did some in silico experimental work to show that by mixing reads from different samples, we could get similar results. And we showed that we could get 95% classification accuracy with only 10 molecular barcodes being detected per marker, or 15 molecular barcodes being detected per marker for the 24 marker panel and the 6 marker panels respectively. Having defined those quality control criteria, it therefore means that the assay could be deployed in the clinic and a genetics team would be able to identify any unreliable results. We also showed that the assay was reproducible and portable. We did repeat testing of 32 colorectal cancers and found 100% concordance and high correlation between the results of the previous test and the repeat test. We then also showed that we could deploy the assay into the local hospital, and on a relatively small cohort, although we have analysed now well over 100 samples in the Newcastle Hospital's laboratories, we showed that we could get, again, 100% concordance or 100% diagnostic accuracy. What was interesting as well was that we showed that 3 of these samples analysed in the Newcastle Hospital had relatively poor sequencing, but they were still classified accurately, which was in line with the in silico predictions. So as a summary, we have an assay that can detect microsatellite instability using next generation sequencing with very high accuracy using as few as 6 markers. And the performance between the 2 different marker panels, either the 24 or the 6, is relatively similar. And most importantly, the reagent costs for these assays are very low. So even with the 24 marker plus BRAF panel, we can the cost of that is less than £20 per sample. So for future work and what we're currently working on, we want to commercialise the assay, and we're working with Cancer Research UK commercial partnerships to do that. We are getting the assay accredited in our local NHS diagnostic labs, but also in the genomics laboratory hubs in several sites across the UK. And all of this work was done in colorectal cancers. So we're doing more work now to show that the assay can be applied to cancers of other tissues, in particular the endometrium, as endometrial cancers are the other very high-risk cancer in Lynch syndrome. These are just two of the references for the assay. And I would like to thank the Barber Foundation, who funded my PhD, in which I did this work, my PhD supervisory team, who are also the principal investigators of the lab that I'm still working in, Mike, Maro, and John, then my colleagues Christine and Hirsch for all of their help during my PhD. And then additional thanks to the CAP3 clinical trial team and the Northern Genetics Service, who have been a support throughout this. And thank you for your attention.
Thank you ever so much, Doctor, for that. I'm hoping that we get the maximum benefit from the information you've presented, since Iraq is actually one of the countries that recently announced the data from Iraq's National Cancer Registry, that there are more than 31,500 cancer and tumor-related cases in Iraq, which is an alarming figure. So the information you presented is highly actually important to us, and thank you ever so much for that.
No problem, my pleasure. Thank you.
Thanks a lot, thanks. And now, last but not least, Dr. Nisreen Al-Hamoud, Director of the Center of Excellence in Biosafety, Biosecurity, Biotechnology at the Jordan Royal Scientific Society. Research emphasis the preservation of human health and biodiversity. Dr. Nisreen, are you with us?
Hello. Good morning. Good
Hello,
afternoon.
Doctor. Good morning, darling. How are you?
Can you see my screen?
Yes, I can see your screen now.
Okay.
Okay, you may start.
Sorry?
You can start now, Dr. Nisreen.
Yeah, okay, yeah. Good morning, صباح الخير. Good afternoon. I'd like first to thank the Qadisiyya University, the organizer of the conference, to invite me to speak in this conference. Very interesting, you know. And actually, the previous three keynote speakers gave a very interesting, very impressive presentation. Especially, I'd like also to thank Dr. Mohammed Al-Askari, a colleague that I know for a couple of years now. Thanks a lot for your invitation, and I wish you all the best. I will speak about something different. I will speak about risk assessment and control systems of genetically modified organisms. First of all, we speak about, you know, international regulations and guidelines and any compliance procedures internationally regarding genetically engineered organisms. Then we speak about the hazard associated with this technology and recombinant DNA. The last thing we speak about risk assessment and management related to procedures for conducting and working with genetically modified organisms. Recombinant DNA molecules is constructed outside the living cells by joining natural or synthetic DNA segments with DNA molecules that can replicate in a living cell. So this is what we call recombinant DNA molecules. There are many guidelines for research, including and involving recombinant DNA molecules. And all these documents are related to principles for the safe conduct of research related to DNA technology. So they detail the practices and procedures for any research related to recombinant DNA research, any proper conduct of research involving GMO, GM plants and animals, and also safe conduct of human gene transfer research. When we speak about is it safe or not, is DNA, recombinant DNA technology, is safe or not, we have two opposite trends of thought. One is US, Canada based, and other is internationally European based. For US and Canada, they say that there is no risk associated with GM crops and no extra regulation are necessary. And they do safety assessment for product-based rather than process-based. So they do have adopt the principle of familiarity and substantial equivalence to conventional crops, where the concept of familiarity has been used in chemical industry to determine safety levels on the assumption that closely related chemicals will behave in the same way. This approach is now widely used in Canada and US to GM risk assessment, genetically modified organisms. Such models have a high level of appeal because they don't require regulators to deal with complex and case-specific factors. This framework neglects the issue of magnitude and rare but significant impact. It may not be well suited to living modified organisms as they can behave in an unpredictable way. The substantive equivalence between organisms is used as an indication on how they will behave, how genetically modified organisms will behave. The concept was originally developed as a way for determining food safety. If a new GM product is substantially equivalent in chemical composition to its natural antecedent, then it is assumed to be safe to be consumed. This approach neglects the uncertainties around the actual modification of DNA. A GM food will be considered to be substantially equivalent to its natural product if after a comparison of several different characteristics, no difference is shown. So US and Canada, they adopt these two concepts when they are working and dealing with GM cases. In comparison to EU, they say that EU crop, genetically modified organisms or genetically engineered crops are considered new and special, and that the existing legislation is not considered sufficient to evaluate the safety of food, genetically modified food to be consumed. So they ask and they request safety assessment based on the process, not on the product. So they want to assess the whole process for producing a genetically modified plant or genetically modified organisms or genetically modified animal. So principle of substantial equivalence at the beginning, they adopted rather than the end. So they adopted at the beginning, then they do assessment for the whole process to produce a genetically modified and engineered product. And they adopt the precautionary principle as a guide for the assessment of GE crops, where scientific information is insufficient, inconclusive, uncertain, and there were an indication or prediction that possible effects will be on the environment, on the biodiversity, on the human, on the animal, on the plant, then there should be an action to protect and to avoid the negative impact of this product. So there are, DNA technology carries certain inherent unpredictability. We can say that unexpected substance may appear during the process for producing genetically modified and engineered food. GM food approved on the basis of substantial equivalence that it is adopted by Canada and US are not safe because they are not tested rigorously enough. And safety assessment based on substantial equivalence is not scientifically based. This is according to the EU regulation. So when we speak about genetically modified food classification, it may be consists of living organisms like, the mostly GM food, soybean and maize, food derived from GMO like soy oil and corn flour, or food containing ingredients produced by genetically modified organisms like some vitamins or essential amino acids, and finally, foods containing ingredients processed by enzymes produced by genetically modified organisms like high fructose corn syrup produced using recombinant glucose isomerase. So there are different classifications when we are trying to assess the safety of genetically modified organisms' food ingredients. When we say about and we want to determine the safety issues in transgenic crops, we speak about how it impacts the biodiversity, the environment, the human, the animal, health consequences. Is there any consequence on the human and animal health? We should assess the short and long-term impact of using these organisms. And biosafety risk involve the entire spectrum when we speak about biodiversity because it is linked to the Cartagena Protocol. And the universal true for all approach is not applicable when we speak about genetically engineered crops. There are many benefits and controversies regarding genetically engineered food. When we speak about crops, maybe it will give you a high yield, good quality, high nutritious food, stress tolerance to water stress, to salinity, but on the other hand, maybe there are potential on human health impact, like producing allergy, transfer of antibiotic resistance markers, also another potential environmental impact related transfer of transgenes through cross-pollination. So there are benefits, pros and cons related to using and handling GM products. For animals, maybe it will yield good yield of meat, eggs, and milk. But when we speak about the access and intellectual property, there will be a domination of world food production by a few companies in the whole world. When we speak about environment, there will be less use of herbicides and pesticides, but at the same time, worldwide, there are really few countries in the world who mandate the use of labeled GE or genetically modified food on the foods, that the consumers has the right to know if his food or her food is genetically modified or not. And of necessity, we speak about food security. This is a new trend of food security, how to secure food for the increasing number of population worldwide. Maybe we can benefit from the advancement in genetically modified products, but at the same time, these new advances will be skewed by the interests of rich countries in the world. When now we want to do risk assessment of genetically modified organisms, the main objective is to have safe and plenty supply of food. But at the same time, we want to preserve the ecosystem, the biodiversity. We need to find a balance between production and wise stewardship of the earth. So this is the main objective why we are going to do a risk assessment for genetically engineered and modified crops. Risk management, we need to have regulation, legislation related to genetically engineered crops. We need to have, there should be a demand on scientific and political vigilance on these products. We need to have oversight on case-by-case, on step-by-step basis, not only, the products, the final product. We need to study and assess every case that we have a new product or new crop that we develop. And we don't support a ban on all GMOs. We need to study every case, every new genetically modified organism product and see if it has an impact, positive or negative, on the health of a human, the animal, and the environment. Risk communication, also, we need to find this, like fear, from consuming genetically modified food and crops. We need to find, public communication tools in order to bridge the gap between scientists, politicians, and the public. We need to have two-way flow of understanding between the scientists and public. We need to not just one-way communication, but encourage dialogue among all participants, among all stakeholders. Everyone has the right to mention and his point and his fear, and how we can address these fears from the public and tell them that there are benefits, there are some controversies, and we can, through risk assessment and communication and management, we can minimize the risk and have the benefit from applying genetically engineering on crops. So risk assessment is the process of identifying the hazards on the human, the environment, through planned release activity. When we are speaking about risk assessment, there should be not unintentional release to the environment of these genetically engineered crops, but should be a planned release activity. So through the risk assessment, we'll identify the hazard on the human, on the animals, and on the environment. We can assign the magnitude and the possibility or probability of adverse effects. And this all concepts, the whole concept of risk assessment, is in line with the Cartagena Protocol on Biosafety, where we should identify and evaluate the potential adverse effects of living modified organism on the conservation and sustainable use of biological diversity and biodiversity in the likely potential receiving environment, taking into consideration the risks on human health. So when we speak about, what are the components of risk assessment? First, risk identification, identifying the hazard. Risk evaluation and risk mitigation. Risk identification, what possible danger is expecting from using and consuming and handling genetically modified organism. Well, we want to do a comparison between consuming GM product and its traditional counterpart. Risk evaluation, what is the probability that such danger will occur? How much damage can be expected from such occurrence? And how does it compare to its traditional counterpart? Then when we identify the hazard, the magnitude of the danger, and to evaluate this danger, we said, can we have any countermeasures and risk mitigation measures in order to reduce the probability that the danger will occur and to minimize the damage if such a danger occurs? So we have, when we speak about risk assessment, we have known probability and unknown probability. Some hazards are identified through the assessment process, and some hazards are not, well identified. So we need to have a rigorous scientific assessment. We need to have risk mitigation based on countermeasures that are improbable to the risk identified, and we need to adopt the precautionary principle that is adopted by the EU. When we speak about, different trials, we speak about lab, greenhouse stage, and confined trial stage. We started with, two main stages, one inside the lab, in order to have, laboratory experiment, laboratory trial, and then we go to the field, confined trial stage. But one important issue to guarantee before, working with genetically engineering organisms is to prevent the spread of genetically engineered material outside the lab and field. So it should be a confined, contained place in order to conduct genetically engineered experiments and trials in the field. Because of this, we need specific biosafety levels and biosafety level facility in order to conduct such a work. So if we are working with genetically modified organism with risk group 1, it should be like contained in containment level 1. If it is like higher, then risk group 2, containment level 2. 3, more dangerous, we need to do it in containment level 3, and serious dangerous, it will be in containment level 4, according to the risk group and according to the activity that we want to apply in this group of organisms. So then we go from the lab, we did our first experiment, we modified the organism, then we want to do a trial in the field, what we call a confined trial in the field on a small scale to test the crop performance when it is released to the environment. So we need to do a confined trial in a small scale release of transgenic plant species for research purposes, because we modified the plants inside the lab. Now we are going to do a trial in the field. So we want to release it on a small scale, on a confined scale, in order to prevent the spread of the organism and mitigate its impact on the surrounding environment. So the objective of doing a confined trial in the field is to evaluate the crop's performance when it is intentionally released to the environment. So field trials on GM crops started 16 years ago. It started in 2004 when more than 170 million acres of GM crops planted. And at that time, more than 25,000 field trials conducted on more than 60 genetically engineered plants in more than 45 countries around the world. And most GM products and GM crops are soybean, corn, maize, cotton, canola, squash, and papaya, which are herbicide and pesticide resistant. And because of this, pesticide use in this country reduced by 2.7 million pounds, and 90 million fewer sprays of herbicides were less. So it's improved the yield, improved the health of farmers from exposure to these herbicides and pesticides, and also at the lower cost. But at that time, the Greenpeace, especially in Europe, opposed the use and the production of genetically modified organisms and crops. So what are the first steps in risk assessment? When speaking about first step, we said, okay, our first step is what we call setting the scene for risk assessment. What is setting the scene? And then when we set the scene, everything is ready to conduct an actual risk assessment. So the first step is risk assessment. It is useful to have what we call, a list of things that we are going to do before starting with the actual risk assessment. So who's the applicant? Who wants to apply this technology in his lab and his field and then to release it into the environment? The recipient organism, including whether the recipient plant can cross-pollinate and cross-fertilize with other natural and wild flora and fauna and cultivated crops, is very important. If it is like very close taxonomically to other plants in the area. And then the type of application, it is contained used, only we want to do a lab work, or we want to go further to do a field trials under controlled conditions, or the last stage, which is commercial release. And what are the inserted or modified genes or sequences? So this is very important. Before you are conducting a risk assessment for any new and genetically modified organism, we need to know all this information before give the startup of the project. The actual risk assessment, it should be identify, like we mentioned earlier, potential adverse effects. Is there any potential adverse effects that may arise from the modification of the phenotypic or the genotypic characteristics of the GMO and its introduction into the receiving environment, taking into consideration, according to Cartagena Protocol, the human health and also the health of the animals, the likelihood that such an adverse effects will take place, and what are the likelihood of potential receiving environment? What are the likelihood of consequences of the potentially receiving environment of this newly modified organism? What are the consequences that would arise from this likelihood of having this danger? And do we have any appropriate risk management strategies that we can apply in order to minimize the risk and the adverse effects? And finally, we need to estimate the overall potential environment impact, including we need to make a comparison between the cost of doing this technology, any potential environmental impact, and any beneficial impact on the human health and the environment. So we need to compare what are the risks and what are the benefits from conducting such experiments and such trials in the field. And if the benefit exceeds the negative impact, then we can tell the researcher, go ahead and start your experiment. Discussing procedures, what are the procedures for GMO to be used for field trials? We need to have core questions. When I go to assess any new product, any new genetically modified product in any research lab or university, I need to ask the researchers four questions. What is the objective of your proposed activity? What is the intended eventual use of the organism to be released? What are the species that you are going to modify? Is it capable of doing any problem, any disease to the human, to the environment, to the animal? It has any, potential impact on the biodiversity surrounding the research area? Please describe the site chosen for the planned release. How close is it, to the populated area? To any centers of agriculture activity and protected area? Is there any endangered species surrounding the research area or no? Habitat and ecology. What is the natural habitat of the wild type organism? Is the wild type organism or its closest taxonomic relative present in the near sites of the research area or no? GMO genetics, what genetic manipulation are going to apply? What is the characteristic of the GMO? How it differs from the wild type counterpart? How many copies are we going to manipulate? And copies of manipulated gene DNA sequence will be present after the experiment. How is the level of expression regulated? Data on stability, survival, and transfer under contained conditions inside the lab. Was the organism studied under containment, in a confined environment? What is the containment level that was used in studying this organism? What are the dispersal mechanisms to disperse this organism in air, water, and soil? Was it studied carefully or not? Finally, I will ask for in detail experimental procedure, the overall experimental design for the release, including methodology, layout of the site for release, schedule of release, and duration of the entire activity. Now, what is when we have like one after the confined and contained experiment in the lab, now we are going to do a trial in a confined site. So what is the field evaluation on the indicative conditions of the proposed test site? First, we want to consider the physical environment, the area, the soil type where the crops will be planted, the climate condition, meteorological data, rainfall pattern, wind direction, velocity, temperature, relative humidity. All these physical environment should be taken into consideration when you choose the site for the field trial. Biological environment, vegetation, any vegetation in the area, entomological communities, is there any native insects, reptiles, and avian species in the area, profile of the microorganisms that are in the site area. Finally, the social environment. Is it close to local population? Is it close to any economic activities? To any health facilities? To hospitals? What is the security of the site? So when I choose the site for confined trials, I need to take into consideration all these requirements before I initiate my confined trial in the site. And this is according to the risk assessment. After we finish all this risk assessment, with the setting the scene and the actual risk assessment, we go and focus on risk mitigation, which is the terms and conditions that are necessary to conduct the trial safely in the field. So after we finish the risk assessment in the lab, in the site, then we want to do a risk mitigation for the hazards that we identified in the risk assessment process. So we want to prevent gene flow, we want to prevent persistence of GMO in the field after harvesting, and we want to prevent entry of GMOs into the field chain. So we have 3 P's of risk mitigation, 3 P's. Prevent pollen-mediated gene flow, gene flow through pollination. We need to do an isolation according to the space. We need to isolate the area for conducting the confined experiments with other, there should be a buffering zone. And this is also according to Cartagena protocol, buffering zone between, the experimental site and other agriculture activities in the area. We need to isolate according to the time-based temporal isolation, and also we need to do physical isolation, tenting and detasseling, like removing the pollen producing flowers in order, it's a form of pollination control employed to crossbreed or hybridize 2 varieties of species. So we need to prevent first gene flow. We need to prevent the persistence of the genetically engineered plants in the environment through monitoring of land use after harvesting. And also we need to prevent the introduction of genetically engineered plant material into the food chain. So we need to do monitoring, controlling the off-site movements after harvesting. We need to control the storage of genetically engineered crops and the disposition of these new products. So when we speak about risk assessment for human health, we need to focus on very well-known international guidelines by Codex Alimentarius Commission. This is Codex Alimentarius, it's mean in Latin, book for food. It's a collection of internationally recognized standards, codes of practice, guidelines, and other recommendations related to food production and food safety. And in this, guideline, they focus on assessment of possible allergenicity, assessment of possible toxicity, analysis of composition of key components in the food and food processing, the whole food processing, and nutritional modification. So this is, one guidelines that it focuses on the production of food and its safety. When it comes to legislation, most legislation and regulation call for case-by-case and step-by-step evaluation of GMO. And this is based on the EU Directive 2001 and based on Cartagena Protocol. They don't want only to test the final product, as it is, the procedure followed in Canada and in US. They want to test and to assess the risk on every case and on every step in the process. And they do want to do environmental and impact assessment that includes effects on target and non-target organisms. So according to the EU directive, they want to evaluate short and long-term impact of this new product. They want to see the direct exposure of the impact of direct exposure of this new product, and also the indirect exposure through chain of events and through the food chain. However, there are, until today, no approved methodologies and best methodology in order to violate all the requirements that it is mentioned in the EU directive. Unfortunately, we need an organization similar and equivalent to the Codex Alimentaris. Until today, we don't have an organization that can, work on food assessment related to genetically engineering products. We don't have this organization until today. But we have international players on this domain, like the Organization for Economic Cooperation and Development, or OECD. We have the International Plant Protection Convention, and definitely we do have the Cartagena Protocol on Biosafety, which is, an agreement which is signed by, most countries in the world. Controlling risk of work with GMO, the purpose of GM risk assessment process is to enable us to select the most appropriate control or combination of control measures, which is appropriate to the level of risk and to the identified hazard. Control measures are systems and action used to reduce the risk of exposure to genetically modified organisms. And these control measures and mitigation measures include engineering controls, such as containment lab, biosafety level 1, 2, 3, and 4, and biological safety cabinet. So this is called, we can include them as engineering control. We have the management controls as standard operating procedures in the lab and also in the field, should be, well developed and followed by the researchers. We do have training before using these organisms and supervision from your mentors and your supervisor and your professor. And also you need to wear PPE, personal protective equipment like lab coat, gloves, and spectacles. Control measures will, reflect the risks from using these genetically engineering techniques, the activities that will be implemented, and any potential route of exposure of people, animals, plants, or release to the environment. So the risks involved should be, based on systematic approach that require the application of the most effective measures, which are reasonably practical and inappropriate to the risk, identified through the risk assessment. And we should, apply the hierarchical approach, where the most effective control measures must be used in preference to the least ones. So we start with the elimination, followed by substitution of the hazard or the risk. Then we go to engineering controls, administrative controls, and lastly, PPE. Once you have decided that we cannot eliminate the hazardous activities, or we cannot substitute the hazardous activities with less hazardous ones, then you should implement control measures that prevent or minimize the exposure to the risk. The control measures must be selected in this order of priority. So in the context of biosafety measures, based on a risk assessment, we can do like wild-type biological agents or genetically modified organisms are assigned to one of the 4 risk categories. So you apply risk assessment to the wild-type biological agents and to the genetically modified organisms. And according to this assessment, you can say that it is according to category 1, 2, 3, and 4. And based on that, you want to perform it in a containment measure. So you can do it in containment lab 1, 2, 3, and 4, according to the safety of this group of microorganisms. Risks are contained by a set of measures employing biological and physical barriers. And the physical containments, it's gradual, physical barriers from lab practices, management, primary containment, to lab equipment doing in biological safety cabinet, and lab facility containment level, biosafety level 2, 3, and 4. So this is like a gradual level of protection from primary containment to secondary containment. Regarding biological containment, it's according to the donor, to the host, and to the vector, for these microorganisms. So in conclusion, safety assessment protocols, there are 2 protocols, international protocols that are adopted by two parties in the world: substantial equivalence, which is adopted by US and Canada, and precautionary principle, which is adopted by the EU directive and the Cartagena Protocol, who adopt this precautionary principle for using genetically engineering techniques. Risk analysis must be undertaken to predict the occurrence of any negative impact on the environment or on the health of human and animal. According to these assessments, we can have like a predictive measures how to mitigate or to avoid the adverse effect that can result from potentially or identified hazard. Risk assessment need to be done for the whole process, case by case, step by step. Should need to be performed on the host and on the donor material. And we need to do like a cost-benefit analysis. Case by case, you should conduct a cost-benefit analysis. Is it beneficially and costly benefit to conduct this research or not? And based on this cost-benefit analysis, which is applied to case by case, then we can decide to go ahead with this experiment or stop it in the lab or go further to confined trial in the field or to the intentional release to the environment. According to the risk assessment, appropriate control measures must be developed, initiated, and followed when performing recombinant work. And thank you for your attention.
Thank you very much, Doctor, for the insightful lecture. Thanks a lot for your time, and thank you for being patient. Thank
Well,
you.
Pleasure.
I would like to, thank you. I would like to call Dr. Mohammed Al-Askari, the Dean of the College of Biotechnology, to say a few words before we end. تفضل دكتور محمد. دكتور محمد ويانا. دكتور محمد. دكتور أحمد تسمعني؟
تفضل دكتور، حول كوهوست. دكتور
محمد موجود؟
لك الصلاحية دكتور.
أعتقد انقطع النت ورجع مرة ثانية. موجود؟
لا رجع دكتور. لا كان يرجع دكتور محمد. تفضل دكتور. تسمعني دكتور محمد؟
دكتور محمد ويانا؟
اكو مشكلة؟
انقطع عنده ورجع.
دكتور سوي خروج ورجع من جديد.
بس دكتور محمد حب يشكر الكينوت speakers. He wanted to thank the keynote speakers himself. So thank you for being patient.
هو موجود حاليا. العفو دكتورة، لحظة بس خلي.
نعم.
راح يرجع إن شاء الله.
تمام. الو دكتور احمد موجود؟ دكتور محمد لو انهي موجود دكتور محمد رجع
وياكم
عيني دكتور عودا حميد ان شاء الله.
شكرا.
تفضل دكتور المايك إلك. تفضل دكتور.
Dear keynote speakers, dear Dr. Kadhim Mohammed Ibrahim, it was fantastic lectures that you made about the advances in blood technology. We got a lot of information and maybe promising ideas to apply it in Iraq in the future. By the way, Dr. Bilal Kamil Suleiman Al-Waili, my colleague, when we were in postgraduate studies, you made such fantastic lecture and presentation about the human genome and genetic variation and genomic technologies that could open the road for our lecturers and our researchers in Iraq. Also, Dr. Richard Gallan, with your presentation about the application of next generation sequencing to hide it through microsatellite instability testing. Dr. Nisreen Al-Hamoud, you are my instructor and I learned a lot by your hand about the biosafety and biosecurity back in Jordan. Thank you for your wonderful lecture that you made about the risk assessment and control system of GMO. And on behalf of Presidents of the University of Al-Qadisiyah, Professor Dr. Kadhim Al-Jaburi, the Deanery of the College of the Biotechnology, conference committees, attendants, please accept our sincere appreciation for the outstanding presentation you made to the Biotechnology Conference, which is your conference, about your highly leveled experiences and organized valuable information you gave. It was very interesting to all of us to hear about your experience and results. Thank you so much for sharing your time and your experience with us. Hopefully to see you once again next year with a new scientific events. From our hearts, thank you so much for being with us. Thank you. Dr. Asma.
Thank you very much, Doctor. Now we'll have a break till the afternoon session, which will resume at 2 o'clock, chaired by Assistant Professor Dr. Wafa Al-Kabi. محاضرات بعد الظهر ستبدأ في تمام الساعة الثانية بعد الظهر إن شاء الله، وحسب الجدول الذي سيعرض الآن على الشاشة، لذلك نسترعي انتباهكم. تحياتي وأمنياتي بالتوفيق للجميع. أسماء عبد الأمير الرئيس، مدير قسم البعثات والعلاقات الثقافية في رئاسة جامعة القادسية. شكرا جزيلا لكم. Thank you. أستاذ أحمد، الجدول الآن جدول بعد الظهر والبريك. تحياتي لك. شكرا جزيلا دكتور أحمد.
السلام عليكم جميعا، ونرحب بالسادة الباحثين والمشاركين والداعمين لمؤتمرنا الدولي الافتراضي الأول في التقانات الإحيائية، وتحت شعار ببحثنا العلمي نواجه جائحة كورونا المستجدة، حيث المناخ العلمي لتبادل الأفكار ونتائج البحوث العلمية الرصينة لخدمة المجتمعات. أهلا وسهلا بكم في هذا المحفل العلمي البهيج، وشكرا لكم جميعا. محمد عبد الوهاب العسكري، عميد كلية التقنيات الإحيائية في جامعة القاهرة. Assalamu alaikum. We welcome the researchers, participants, and supporters of our first virtual international conference of biotechnology under the slogan, "Our scientific research challenges the COVID-19 pandemic," to exchange the ideas and scientific research results to serve the societies Once again, you are most welcome in this gorgeous scientific forum. Thank you. Mohammed Abdul Wahab Al-Askari, Dean of the College of Biotechnology, University of Al-Qadisiyah.
الشمال الغالي يبقى شامخ عالي يسمو كالجبال كاكدما والوسط قريبة والغربية حبيبة والفرح تجيبه باسم العراق كل العراق والجنوب يوافي والنخل عوافي ويا قلبها الصافي نبض العراق بيت كل أهلنا وصوتنا وآملنا بصوت عالي كلنا نريد السلام كل العراق زاهية وحبوبة ونادرة وطيوبة فيها بكل عذوبة هالقلبها سالمة يا بلادي يا تراث أجدادي ويا رمز بغدادي وعاش العراق يسحر النواظر ويسر الخواطر بين ماضي وحاضر دار السلام يا بلادي سيري مصيرك مصيري والوفا ضميري لأهل العراق عراق عراق كل العراق
السلام عليكم جميعا، ونرحب بالسادة الباحثين والمشاركين والداعمين لمؤتمرنا الدولي الافتراضي الأول في التقانات الإحيائية. السلام عليكم جميعا، ونرحب بالسادة الباحثين والمشاركين والداعمين لمؤتمرنا الدولي الافتراضي الأول في التقانات الإحيائية، وتحت شعار ببحثنا العلمي نواجه جائحة كورونا المستجدة، حيث المناخ العلمي لتبادل الأفكار ونتائج البحوث العلمية الرصينة لخدمة المجتمعات. أهلا وسهلا بكم في هذا المحفل العلمي البهي، وشكرا لكم جميعا. محمد عبد الوهاب العسكري، عميد كلية التقانات الإحيائية في جامعة القادسية. السلام عليكم. We welcome the researchers, participants and supporters of our first virtual international conference of biotechnology under the slogan "Our scientific research challenges the COVID-19 pandemic," to exchange the ideas and scientific research results to serve the societies. Once again, you are most welcome in this gorgeous scientific forum. Thank you. محمد عبد الوهاب العسكري, Dean of the College of Biotechnology, University of القادسية.
الشمال الغالي يبقى شامخ عالي، يسمو كالجبال كاكا تمام، والوسط قريبة والغربية حبيبة، والفرح نجيبه باسم العراق. كل العراق والجنوب يوافي، والنخل عوافي، ويا قلبها الصافي نبض العراق. بيت كل أهلنا وصوتنا وآملنا، بصوت عالي كلنا نريد السلام. زاهية وحبوبة ونادرة وطيوبة، فيها بكل عذوبة هالقلبها. سالمة يا بلادي يا تراث أجدادي، ويا رمز بغدادي وعاش العراق. يسحر النواظر ويسر الخواطر، بين ماضي وحاضر دار السلام. يا بلادي سيري مصيرك مصيري، والوفا ضميري لأهل العراق. عراق عراق كل العراق.
السلام عليكم جميعا، ونرحب بالسادة الباحثين والمشاركين والداعمين لمؤتمرنا الدولي الافتراضي الأول في التقانات الإحيائية، وتحت شعار ببحثنا العلمي نواجه جائحة كورونا المستجدة، حيث المناخ العلمي لتبادل الأفكار ونتائج البحوث العلمية الرصينة لخدمة المجتمعات. أهلا وسهلا بكم في هذا المحفل العلمي البهي، وشكرا لكم جميعا. محمد عبد الوهاب العسكري عميد كلية التقنيات الإحيائية في جامعة القادسية. السلام عليكم. We welcome the researchers, participants and supporters of our first virtual international conference of biotechnology under the slogan Our Scientific Research Challenges the COVID-19 Pandemic, to exchange the ideas and scientific research results to serve the societies. Once again, you are most welcome in this gorgeous scientific forum. Thank you. محمد عبد الوهاب العسكري, Dean of the College of Biotechnology, University of القادسية.
الشمال الغالي يبقى شامخ عالي يسمو كالجبالي كاكا تمام، والوسط قريبة والغربية حبيبة والفرح نجيبه باسم العراق. كل العراق والجنوب يوافي والنخل عوافي ويا قلبها الصافي نبض العراق. بيت كل أهلنا وصوتنا وآملنا بصوت عالي كلنا نريد السلام. زاهية وحبوبة ونادرة وطيوبة فيها بكل عذوبة هالقلبها. سالمة يا بلادي يا تراث أجدادي ويا رمز بغدادي وعاش العراق. يسحر النواظر ويسر الخواطر بين ماضي وحاضر دار السلام. يا بلادي سيري مصيرك مصيري والوفا ضميري لأهل العراق. عراق عراق كل العراق.
السلام عليكم جميعا، ونرحب بالسادة الباحثين والمشاركين والداعمين لمؤتمرنا الدولي الافتراضي الأول في التقنيات الإحيائية، وتحت شعار ببحثنا العلمي نواجه جائحة كورونا المستجدة، حيث المناخ العلمي لتبادل الأفكار ونتائج البحوث العلمية الرصينة لخدمة المجتمعات. أهلا وسهلا بكم في هذا المحفل العلمي البهي، وشكرا لكم جميعا. محمد عبد الوهاب العسكري عميد كلية التقنيات الإحيائية في جامعة القادسية. السلام عليكم. We welcome the researchers, participants and supporters of our first virtual international conference of biotechnology under the slogan Our Scientific Research Challenges the COVID-19 Pandemic, to exchange the ideas and scientific research results to serve the societies. Once again, you are most welcome in this gorgeous scientific forum. Thank you, Muhammad Abdul Wahab Al-Askari, Dean of the College of Biotechnology, University of Al-Qadisiyah.
الشمال الغالي يبقى شامخ عالي يسمو كالجبال كاكا تمام والوسط قريبة والغربية حبيبة والفرح نجيبه باسم العراق كل العراق والجنوب يوافي والنخل عوافي ويا قلبها الصافي نبض العراق بيت كل اهلنا وصوتنا واملنا بصوت عالي كلنا نريد السلام كل العراق زاهية وحبوبة ونادرة وطيوبة فيها بكل عذوبة هالقلبها سالمة يا بلادي يا تراث أجدادي ويا رمز بغدادي وعاش العراق يسحر النواظر ويسر الخواطر بين ماضي وحاضر دار السلام يا بلادي سيري مصيرك مصيري والوفا ضميري لأهل العراق عراق عراق كل العراق
السلام عليكم جميعا ونرحب العلمي نواجه جائحة كورونا المستجدة، حيث المناخ العلمي لتبادل الأفكار ونتائج البحوث العلمية الرصينة لخدمة المجتمعات. أهلا وسهلا بكم في هذا المحفل العلمي، وشكرا لكم جميعا. محمد عبد الوهاب العسكري، عميد كلية التقنيات الإحيائية في جامعة القادسية. السلام عليكم. We welcome the researchers, participants and supporters of our first virtual international conference of biotechnology under the slogan "Our scientific research challenges the COVID-19 pandemic," to exchange the ideas and scientific research results to serve the societies. Once again, you are most welcome in this gorgeous scientific forum. Thank you, Muhammad Abdul Wahab Al-Askari, Dean of the College of Biotechnology, University
Hello? Hello?
دكتور الصوت واضح.
صوت واضح. Thank you so much. And on behalf of the University of Al-Qadisiyah, I do welcome the president of the Al-Qadisiyah University, Professor Dr. Kazim Jabr-Jaburi, and all of you, our colleagues, researchers, attendants for this scientific forum, the first virtual conference of biotechnology. And I do welcome Gwen Kaut, the manager of CRDF Global, which is organization concerned about the biosafety and biological hazard in Iraq and overall the world. Once again, welcome all of you. And hello, Mrs. Gwen. You can share your slides. Please.
Can you see my slides now?
Yeah. Go ahead.
Good. Thank you so much for inviting me for this very important conference in time of COVID-19. I'm very honored to be able to talk with you today about the work that CRDF Global did and is still doing in Iraq, thanks to our partnership with so many of you in Iraq. So my name is Gwen Kwat, and I work at CRDF Global, particularly focusing on the Iraq program at the CBRNE division in our headquarters located in the United States in Virginia. So CRDF Global was authorized by Congress in 1992 and established in 1995. So we are actually celebrating our 25 years of existence this year. And we have approximately 200 staff worldwide, and we are operating in over 40 countries. The Congress established us in '92 because in the time of the fall of the Soviet Union, so that we could work with the former Soviet Union and newly states created after the fall to work with the scientists so that we can work for a peaceful world together. Today, during the slides and the presentation, we're going to focus on the biological, chemical, and nuclear security, especially, of course, for us, the biological security, as the program in Iraq from CRDF Global is mainly focused on biological security. So this is the global reach for CRDF Global worldwide. You can see that we have a lot of countries we work with throughout the years and the 25 years of our existence. We are located mainly in, our headquarters is in Arlington, Virginia. And we also have a regional office in Ukraine as well as in Amman, Jordan. So what do we do? So as I said, we do have 25 years of history, and we support mainly scientists, industry, chemical industry, biological industry, and law enforcement all around the world to achieve the fullest potential of the countries for securing the dual use research and nature of the work in the laboratory. We partner mainly with international government agencies, industry, academia, and non-government organizations to develop solutions to mitigate the proliferation of chemical, biological, radiological threats. So our mission is for peace and security using science in a way and facilitating the work of scientists. Our projects strengthen international safety and security culture by enhancing risk management practices, improving in-country detection and response capabilities, securing facilities, and encouraging collaborative partnerships to reduce the risk of the research and access to the pathogens in laboratory. So our capabilities is, as you can see here on this slide, it's an overview of what we do. And mainly for Iraq, we focus on human capacity building, risk detection and mitigation, workshop and trainings, curriculum development, security assessments, equipment procurement, and physical security updates. We don't do really travel and event logistics, tailored fellowship and travel grants, especially now, of course, because of COVID-19, nobody really can travel worldwide. And hopefully we will be able to do that soon. So to focus now on what we did in Iraq, I will share a few highlights on, a few highlights of some important projects that were done in the past years. So let's start with the physical security and equipment procurement. This is actually something that is always going on at CRDF Global, and we do have competitions that are run pretty much every year. And those competitions help to secure biological and chemical facilities. It's not only on the biological side. We try to also focus more on the interdisciplinary between biological and chemical research as well, due to, of course, a lot of chemicals are also used in the biological laboratories. So those competitions are open to mainly university professors to be able to do some laboratory risk assessments, physical laboratory security, access control, and of course, train the staff members. So for example, on that picture that you can see here, you can see staff member being trained on how to use the autoclaves that we procured for the laboratory. So mainly the procurement and of equipment that we implement in Iraq is focused, as I said, on chemical and biosecurity equipment, but also some hard equipment like locks, doors, security systems to contain access to hazardous material in the lab that we don't want to be open to anyone, like bad actors, non-state actors, as you know. So we secure the labs, but also secure the locations, the storage locations where dangerous pathogens and hazardous chemicals are stored. So we do that through chemical cabinets, and also, of course, sometimes we through the locks and the doors, we secure specific areas in the laboratory to store those dangerous pathogens, as well as procuring some freezers with locks and other things. As well, we procure a lot of incinerators, and as you can see here on the picture, some autoclaves to be able to help secure samples after they have been used. So we also help develop the new SOPs to incorporate new security equipment and best practices in the lab. And of course, as we said already, train other staff members to be able to use new equipment or understand what BIOS risk management is. Some other project highlights, we do human capacity building a lot in Iraq as well. Two projects that were implemented in the past was the biorisk management train the trainer workshop for Iraq provinces that Dr. Al-Askari actually attended, as well as the promoting biorisk management through a comprehensive curriculum on responsible science. As you can see, it's not only about the dangerous materials and either pathogens or chemicals that we focus on, but truly the human capacity to be able to work to train others on biorisk management. As you can see, the outputs of the train-the-trainer project that we ran, but also understanding what responsible science means when we talk about biorisk management. But not only that, we can also talk about intellectual property and, of course, false data and publication as well, which is very important for science. In the picture here that you can see, it was a partnership during the train-the-trainer workshop that we held at that time in Jordan. And the Jordanian civil defense held a demonstration and training on their capabilities to be able to disrupt chemical or biological events. We do also curriculum development, and again, Dr. Alaskeri was a part of this, and hopefully we will finally be able to continue the work on that. I have some good news for you, Dr. Alaskeri. So we are developing a biosecurity curriculum for priority universities, but mainly we hope that this is going to be used at some point nationally. With Dr. Al-Hamoud that you heard a little bit earlier this morning, and Dr. Abini, we evaluated some biosecurity programs, and we worked truly with our collaborators locally in Iraq to develop an effective curriculum for graduate and postgraduate programs for biosecurity at the academic institutions. We worked throughout Iraq and the KRG ministries as well to present what the outline of this curriculum is, so that you can hopefully one day have access to that during a diploma for young people at the university. Not only So once we secured the laboratories and have experts in the lab to be able to work securely in the laboratory with dangerous pathogens and chemicals, we also focus on how to strengthen the best practices in the scientific institutions. As you know, Iraq has had a lot of turmoils, especially with ISIS, and access to universities and destructions of the laboratory. It's also looking at the promotion of the personnel reliability programs in Iraq, meaning that once you have your staff, you need to be able to not only grow the staff, but also make sure that the staff that you hired is actually not going rogue and working with non-state actors. This is a little bit difficult to work on, since it attached to really difficult issues and sensitivities usually for the academic institutions. But it's something that we are more and more in understanding that we need to be able to understand who we have anywhere in any workspace, to be able to make sure that these personnel are not collaborating with anyone that would do harm in the country. Finally, we cannot talk about the past and what we do for the laboratories and the personnel in the laboratory if we don't talk about what is happening right now with COVID-19. And the highlights I wanted to show today is about what we're doing with mainly the Ministry of Interior, so we can resupply them in very important equipment and chemicals to be able to respond to the crisis. So We did partner with Dr. Ali El Ameri and the Ministry of Interior to be able to procure disinfectants that we are still actually looking at procuring more, some backpack sprayers, and as well as chemical barriers to be able to go around Iraq to secure the country against the disease. We also worked with the Sandia National Lab to put together an SOP to be able to use the equipment. This trainings and equipment procurement is very important, of course, to be able to be better at containing the disease. But not only on equipment and being able to contain the disease, we need also to be able to, again, move forward at better coordination between CBRN teams from the Ministry of Interior, as well as Ministry of Higher Education, with all of the CBRN teams that are created or have been created in the past at the university level. So we're putting together a remote training so that we can train the teams on leadership communication, emergency response, SOP development, PPE usage, and disinfection. We're very excited about this new prospect, as it's going to be a very interactive remote training with, at the end of the training, we'll have a Q&A sessions to be able to talk directly with the SMEs that we hired for this work. Again, when we work in Iraq, we try to always integrate the central government with the Kurdistan government because fighting diseases or non-state actors at the local level means that integration of agencies, they need to work together to be able to be more efficient. And we've been very successful in that, and we're very grateful of the people in Iraq to be working with us to secure the laboratories and other facilities in Iraq. So this is basically what happened in the past, with brief highlights on some projects in Iraq in the past and present. And of course, we're looking at the future as well. I am not going to go in details this time, but hopefully you will see on our website some opening of competitions and other grants so that we can actually continue the wonderful work that we have been doing in Iraq. It is actually very true. All of the work we've done in the past is for one of the country we've worked at is actually the most integrated and the most collaborative with the people on the ground that we've seen, and we're very, very grateful to be working with you. Our main funder for this work is the Department of State at the US, and also sometimes when we're lucky, because we also need to win some money to be able to work with you, the government of Canada. So I'm welcoming any questions. If you have some, please let me know and I will be able to answer right now.
Thank you, Gwen. Thank you for this fantastic presentation that you made. And not to forget, we have, I think, 2 or 3 together projects we were working together before, right, Gwen? I think.
Yes.
Yeah, Gwen. And now we'll open the door for questions. Anybody who has a question, please raise your hand, and we'll open the door for you to ask Gwen directly. أي شخص عنده سؤال، السلام عليكم. أي شخص عنده سؤال بس يرفع لي إيده، يسوي ريز هاند. أكو إعاز موجود بالأسفل، يضغط على ريز هاند حتى نقدر نميزه ونفتح له باب السؤال. تفضلوا.
I guess there's no question.
I think there is no question right now. So I think Galaxy Note, he has a question. Galaxy Note 9. Yeah, you can ask.
Okay. Good evening, everyone. Good afternoon, everyone. And thanks,
Hello.
Dr. Nguyen, for a nice presentation. I'm Dr. Hero from Talahaddin University, Erbil.
Hello, Dr. Hero. How is it with you?
Hello, Doctor. How are you?
I'm fine.
My respect to you, Dr. Muhammad, and for Dr. Nguyen, and for Dr. Nasrin Al-Hamoud.
Yeah,
And
yeah. Thank you.
Thank you, Dr. Hero. Nice to hear you.
nice to hear you, Dr. Nguyen. My question is, Dr. Nguyen, under this circumstance and this situation of COVID-19, what we can do to improve our security and safety in the labs, especially there is lockdown for institutions and colleagues. So what we do to improve the safety and security? Can it did by online and meeting Zoom meeting online like this? Does it help for improving the security and safety? And thanks.
Yeah, I think it's very important to be able to continue the collaboration, right, between everybody in around the world, your collaboration with us and also with Jordan, Dr. Al-Hamoud. And right now, I think it's what is important is capacity building and making sure that everybody understands how to fight the disease itself, but also try to see if some research collaboration can be done so that Iraq is not behind when the vaccine is coming, right? And then at the laboratories themselves right now is really trying to prepare for when everything is going to reopen, because that's when the danger exists. So right now it's trying to have more collaborations between you in Iraq, sharing ideas, sharing capacity and SOPs and what is being done at each university so that when the country reopens, there's no second wave hitting the country.
We hope so.
Okay.
We hope so. There is no second wave. We hope so. There is no second wave at all. Thank you. Thank you, Dr. Hero. Any question?
You, Dr. Muhammad. Thank you.
As long as Gwen here, and she is so difficult to get Gwen on any webinar, but today she was so generous with us. Thank you, Gwen, for being generous with us and sharing your time with us. And
Thank you so much. Thank you so much. I even had time to drink some coffee before coming on, so that was nice.
Oh, yeah, yeah. You told me. Yeah, you told me. Yeah, you told me.
Thank you, and good luck with this conference. It's wonderful to see all of the people sign up, and yeah, I can't wait to hear some summary of the recommendations at the end. Thank you so much for inviting me again.
Thank you. And by the way, the president of the university, Professor Dr. Kazim Al-Jiburi, he is with us right now on live. So
nice.
maybe you say something to Dr. Kazim Al-Jiburi.
Well, thank you so much for everything we're doing and together, and to secure and work with laboratories to safely work together, especially in these dire times right now with COVID-19. It's interesting, sadly, because we're all working to go against pandemic, either naturally occurring or intentionally man-made occurring, and we are actually living one right now. So our work is really key, and collaboration is the best. Please collaborate.
Thank you so much. Thank you so much for being with us. Thank you for your time, Gwen. And I wish you a nice day now back in US. And maybe you see you when reopening US once again and Iraq.
I can't wait. Yes.
We can meet together. Yeah.
Yes, and this time in Qadisiyah, hopefully. Thank you
maybe
so
we will
much.
invite you to Al University. Maybe.
Yes,
Who
thank
knows?
you.
Who knows? Who knows?
Who
Maybe.
knows? Have a good
Thank you so
day
much.
and wonderful
You
meeting,
too.
everyone. Bye bye.
Thank you. Bye bye. Bye bye. Bye bye, Gwen. And now we are going back to the sessions. Dr. Wafa, Dr. Ala, they will take the lead and do the sessions, the rest of the sessions. Dr. Wafa, Dr. Ala. دكتور علاء.
اهلا وسهلا دكتور.
دكتور زياد، عفوا دكتور زياد, he will take the lead. شكرا دكتور زياد.
Okay. Thank you so much, Dr. Muhammad.
I will give you the flag. I will give you. I will hand you the flag, دكتور زياد.
Thank you so much. Thank you so much. اخواني واخواتي اعزائي الباحثين, dear investigators and our colleagues, welcome in the first conference of biotechnology. اهلا وسهلا بكم بالمؤتمر الاول لكليتنا للتقنيات الاحيائيه. على بركه الله نكمل الجلسه المسائيه الاولى, وحاليا ان شاء الله بعد ان كملت دكتور جوين, thank you so much for her, عندنا البحث المقبل اللي هو الدكتوره صفاء رسم عبد الله والدكتور رنا صالح الدفاعي واسيل عبد الرضا ورماح عبد الحسين صباب, بعنوان Study of Cryptosporidium species parasites spread in domestic birds of Diwania gonorrhoeae using nested PCR. موجودة يعني؟ موجودة؟
مرحبا دكتور زياد. مرحبا.
وعليكم السلام. هلا دكتور وفاء.
شلون الصحة؟ شلون الأحوال؟ آسفة على المقاطعة. طبعا نرحب بالمشاركين والحضور الكرام في هذا المحفل العلمي البهي لكلية التقنيات الأحيائية. دكتور, حبيت أبلغك أنه المسؤول عن إلقاء البحث أو الباوربوينت للبحث الأول دكتورة أصيل عبد الرضا سعيد. يا ريت إذا موجودة ترفع لنا إيديها حتى تبلش بالإلقاء.
سوي ريز هاند بلا زحمة.
ريز هاند, وإذا ما موجودة حتى تبلش بمكانها ست نور بالبحث الثاني, أعتقد نور قاسم البحث الثالث.
أي والله, أتمنى يرفعون يسوون ريز هاند على مود. اوكي, هذا سوى ريز هاند جالكسي 9.
منو دكتور زياد راح يبلش حاليا؟
اكو
عفوا اصيل سعيد.
اصيل عبد الرضا. اصيل عبد الرضا سعيد.
اي موجوده, هاي كو هوست موجوده اصلا, موجوده اصلا. نعم موجوده. فتحت الميكروفون, تفتح لنا الميكروفون.
اهلا وسهلا. اهلا وسهلا.
انا افتح الميكروفون الها. اصيل بلا زحمه عليش, نحاول نفتح الميكروفون إليش اذا تبقين, اذا امكن انه توافقين على فتح الميكروفون حتى تقدرين تتكلمين معنا. اصيل سعيد.
دكتورة أصيل, خلي أخابرها على رقمها الشخصي حتى ننبهها. دكتورة أصيل.
الو. نعم الصوت واضح. نعم الصوت واضح.
نعم الصوت واضح, صوت واضح. دكتورة أصيل, دكتورة أصيل, مرحبا بك. انت هسه على الهواء مباشرة في الجلسة. عليكم السلام دكتورة أصيل. إذا أمكن تسوين شير للزوم للسلايدات اللي عندك وتوكل على الله. أهلا وسهلا بك. مرحبا.
بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. نعم دكتور, واضح الشير؟
دكتورة لي هسه ماكو شير.
نعم. نعم دكتور.
اي نعم وصل. ممتاز.
بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. الباحث أصيل عبد الرضا سعيد، كلية الطب البيطري، جامعة القادسية. The title of our research is Study of Cryptosporidium species parasites separate in domestic birds in Diwania governorate using nested PCR. And this study was conducted to investigate the separate of the natural infection with parasite Cryptosporidium species in domestic birds in Diwania city. The results of the current study showed that the total incidence of Cryptosporidium parasites in birds under study without specifier of sex was 43.9% from the collection of total sample, about 205 samples. We found 19 infection sample was infected, examination and diagnosis. The results of our study, which belong to 4 types of study birds, showed that high incidence of cryptosporidiasis was in Gallus domestic as it reached 530.3 and lowest in the Balgon, Illinois, about 60. While the results showed that the infection rate in Milligas gallibova was 44.3 of the total 60 sample. As for the percentage of the Cryptosporidium parasite infection in Anas chorus, it reached about 41.60 of the total 60 sample. We using 2 methods of diagnosis have been used, acid-fast stain method and DNA polymerization. The PCR is more reliable, sensitive, and less time-consuming approach for the diagnosis of Cryptosporidium infection than the microscopical examination, which was very labor-intensive and requires skillful technique. After combining the 2 methods, the results show that the infection rate of microscopic examination is 43.9, about 90 sample positive from 205 in birds, while the total infection rate was 47.5, about 90 sample positive from 14 positive sample. And the nested PCR test, molecular detection of Cryptosporidium oocyst appears after DNA extracted from positive feces sample. It was amplified using nested PCR. Most the sample yielded positive band and were amplified at the size of nearly 578 base pair. Material and method sample were collected and isolated. The parasite ovarian cyst were 16 chicken were collected from local chicken, 16 from Turkey, 25 Balgon, Illinois, and 16 from the Enos Peripheral. The results. Table number 1, the prevalence of the Cryptosporidium oocyst infection and when we examined total number is 205, infection number 19 with percentage 43.9. Table number 2, the percentage and type infection birds of Cryptosporidium and their number. The highly number infection we found in the Gallus domesticus with percentage 53.3, and the low rate of infection we found in the first species Balgo, Illinois, about 60. When we make comparison between the 2 methods we are used, nested PCR and microscopical examination, acid-fast stain microscopical examination, we found different between the results. We found the results positive in the microscopic examination is from the total percentage, total sample 205. We found 19 positive sample with percentage 43.9, while we make missed PCR for the 40 positive sample, we found 90 percentage about with 90 sample positive with percentage 47.5. From the above, this study examined 25 samples, 205 samples of domestic birds using simple method and using modern and specialized method for Cryptosporidium species. The results of this study proved that the birds infected with the cryptosporidiosis are highly rate with 43.9. These results are not consistent with the several studies, including by 70, which found the low infection rate in several types of the birds. After examination of 60 samples of domestic chicken and turkey, the diagnosis revealed that 32 infection and with rate of 53.3 percentage of domestic chicken and turkey, 29 infection and which an infection rate of 48.3 percentage, which is almost than the infection rate recorded by 18 in Algeria. Also, which suggested that Cryptosporidium infection rate were 34 and 44 in chicken and turkey respectively. Maybe this difference in the percentage infection of rates due to many factors, including location of collecting sample, the age of examined host, feeding condition, degree of contamination of the feed, and drinking water by parasites. Out of 205 fecal sample exams, we found 60 samples were collected from ducks are highly positive with the Cryptosporidium species with the percentage 21.6. Also, we found that this result not consistent with the research by 19 in southwestern of Nigeria, and also in the by 21 by in China, which showed that the infection rate was 50.4 and 60.3 respectively. Also, we found the result of the result of this study showed the diagnosis of 25 specimen for number 25 specimen of pieces of the ornamental birds belong of the type of the Valgo Eleonora infection with rate was 60, and this is not consistent with research by 20. This variation in the infection rate can be attributed to the different region and environments from which the sample were collected. Also, in our study, we found Cryptosporidium parasite in feces sample of different birds by PCR using a specific primers designed according to the gene of 18 sRNAs. In the more recent study in China, the prevalence of the Cryptosporidium species has been reported in details in the Quiltric between duck beds and birds and articles by Hani. Also in Germany, when make research about 265 fecal specimens were randomly collected and from farmed poultry in Germany and screened for the presence of the Cryptosporidium species using nested PCR and defining the results indicate the Cryptosporidium parasites are common among broiler and turkey in Germany. Considering the large size of the poultry industry and the large amount of poultry meat it was consumed. In Iraq, the parasite has been diagnosed in many molecular studies for different type birds.
by PCR methods, such as research by one of the boiler chicken in Qadisiyah province. Thank you for your listening, and if there is any question.
Thank you so much.
Thank you,
For
Dr.
your presentation, Dr. Asil, and now open the door for any question or inquiry that you like, or addition or suggestion. So please your raise your hand. رجاء رفع اليد للشخص اللي يحب يشارك بطرح او سؤال او استفسار او اضافه حتى نقدر نميزه ويفتح له دكتور احمد يفتح له الاشاره ويقدر يتكلم بحريته. تفضلوا. إذن أعتقد ماكو سؤال. ماكو سؤال.
نعم دكتور.
ماكو سؤال. دكتورة وفاء شكرا جزيلا دكتورة أسيل، شكرا جزيلا. دكتورة وفاء، من الذي دكتور زياد من الذي بعده في إلقاء؟ من على قائمة الإلقاء من بعد دكتور أسيل؟ لطفا.
لحظة دكتور، أهلا وسهلا. دكتور عندنا دكتور معتز محمد الطائي، دكتور أحمد ياسين الطربلي، وعندنا وسام حسن هادي وعلي صالح. دراستهم a study of antifungal activity of some antibiotics against Aspergillus species and Candida species in Iraqi patients with diabetes mellitus. إذا موجودين خلي يسوون ريز هاند على مود نفتح المايك لهم ونعطيهم إمكانية عرض الشرائح.
زياد.
عيني. نعم.
مرحبا.
اهلا وسهلا دكتور.
اعتقد حاليا ست نور جاهزه. نور قاسم.
ست نور قاسم.
اي يا ريت تخاف الميكروفون ترفعوا عنها.
دكتوره ضمياء موجوده؟ دكتوره ضمياء مكي؟
لا حاليا ما متوفره الست ضمياء.
ماشي. دكتوره نور جاسم خضر، نور قاسم خضر عفوا، واسماء محمد سعد. دراستهم بعنوان Association of HTR1A gene polymorphism with schizophrenia. موجودين؟ خلي يسوي ريز هاند على مود نعطيها صلاحيه.
مسويهم انت كوهوست دكتور؟
بس خلي يسوي ريز هاند، هواي عندنا بارتيسبنتس مشاركين على مود ندبر.
اها
نور اعتقد شفتها. نور قاسم هاي كوهوست. اي تفضلي ست نور. عيني. يلا تفضل.
الثانية.
اهلا.
اي نسمعك. اهلا وسهلا ست نور.
اهلا ست نور.
اهلا بكم.
اهلا بيك. يا هلا.
شرفتينا اليوم وقاع هذا المؤتمر العلمي لكلية التقنيات. يا اهلا وسهلا بيك.
اهلا بيك. هلا.
يا اهلا وسهلا. يلا تفضلي. اهلا.
تمام اوكي. زين الشرائح.
اي شير سكرين من جوا عندك.
ثواني. أنا ما يطلع يشينا على هسه حاليا. اكو سكر.
اي جوا اكو
سلايدات. انا نسوي شير سكرين شير كونتنت.
لحظه شبكه يطلع هسه.
ست نور
تاكدي من ست نور بلا زحمه.
سوي شير سكرين.
شير سكرين.
from PC
سوية
ما ادري يظهر.
النوبة ضغط سكرين.
إيه سويت سكرين. وراها عندي هاي الأوبشن فوتو زوم ماسنجر يعني أشعل الزوم.
ست نور، كيفك ست
نعم.
نور؟ شلونك
الحمد
ست نور؟
لله.
الله يسلمك.
خير.
انا وياش محمد العسكري، عميد كليه التقنيات. ست نور بلا زحمه
وسهلا.
عليك، اشكرك بلا زحمه عليك. افتحي لي البرزنتيشن خليه هو الظاهر على الديسكتوب. البرزنتيشن مالتك الباوربوينت افتحي لي اياه هسه خطوه خطوه راح اجي وياه ثاني.
اوكي
فتحتيه بابا؟ اي هسه شو تسوين؟ تروحي لي تروحين على شير سكرين. روحي على شير سكرين هسه ضغطي لي اياها جوا بالاخضر تلقينها امامك.
اي تمام.
ضغطي لي اياها واختاري
ضغطت عليه.
اختاري ديسكتوب راح تطلع لك عندك. اختاري الباوربوينت راح تشوفينه قدامك شبحي صوره شبحيه.
ما يطلع اي شيء.
حاليا صورتك موجودة. يعني وسهلا بيك ست رنا، شرفتينا.
بس ثاني صار 4 مرات اسوي نفس الحالة انه ما قاعد يطلع اي شيء.
لا هاي صورتك، هاي مو صورتك. احنا
فاهم عليك فاهم عليك. بس انه قلت لي من الشير دا اضغط عليها سكرين ما قاعد يطلع اي شيء.
انت عارضه العرض مال الديسكتوب بابا؟
اي عارضته
موجود قدامك هسه. روحي على شير سكرين، ضغطي شير سكرين، اختاري العرض من عنده راح تطلع هي صوره الديسكتوب. اختاري ديسكتوب وانتظري. شير سكرين اسفل اكو سهم باخضر، شوفين السهم الاخضر بابا؟
اي هو انا بدي اشر عليه.
اضغطيه اشتغل عندك.
اشتغل عندك. اضغط على السكرين ما قاعد يطلع اي شيء.
انت فاتحه الموضوع
دكتور اذا احد ادى المحتوى، دكتور اذا احد ادى من المحتوى من الاساتذه الافاضل.
اوكي دكتور زياد عندك المحتوى؟
دكتور انا راح شو اسمه اشير لها اني هي بس
منك شيء. اي مو اشكال، هذا نحل الموضوع.
ماشي انا راح اسوي شير سكرين الك ست نور وانت كملي شرح.
اوكي ماشي.
ماكو مشكله بابا، ماكو مشكله. ريلاكس وتنحل. ماكو مشكله، ريلاكس وعافيه عافيه. احنا كل احنا وياك بابا. عايكم.
اوكي
عفوا دكتور محمد، اعرض البرزنتيشن موجود عندي مال ست نور.
عرضه مشكله. دكتور زياد
تعرض له وانا اعرض.
ماكو مشكله. دكتور زياد بده يعرض. اوكي ماكو مشكله.
اوكي
تمام.
اي نعم تفضلي بابا.
ماشي. Human 5H1A receptor RS100-42468 polymorphism and schizophrenia. Schizophrenia is debilitating and often chronic disorder affecting about 1% of population and with the typical onset in early adulthood. Schizophrenia is a heterogeneous syndrome with unclear molecular mechanism. The etiology-pathology of schizophrenia is extremely complex and its reasons have not completely comprehended yet. Both environmental and genetic factors are commonly involved in the outbreak of schizophrenia. Symptoms and diagnosis. Symptoms including hallucination, delusion, positive and negative symptoms such as psychomotor slowness, underactivity, depression, and withdrawal. Many differences in brain structure and function have been reported in cases of schizophrenia as compared with control. Although there is considerable inter-individual heterogeneity. The International Classification of Disease, Standard Edition, and Diagnostic and Statistical Manual of Mental Disorder, 4th edition, are used as diagnostic structure that's based on patient symptoms. HTR1A, this gene encodes G protein coupled receptor for 5-hydroxytritamine serotonin and belong to the 5-hydroxytritamine receptor subfamily. Serotonin has been implicated in a number of psychological processes and pathological conditions. The serotonin 5H1A receptor was first 5H receptor to be fully sequenced. 5H1 receptor binding sites are located in the primary and limbic area, notably the hypothalamus and cortical area. The human gene encoding this receptor has been located on chromosome 5. 5H1A receptor subtype may be critical for schizophrenia and depression. Cytogenic location of 5-HTR1A gene is located in the long arm of chromosome 5 band 12 band 3. Blood collection. Blood samples were collected from 40 Iraqi patients affected by schizophrenia from Ibn Rushd Psychiatric Hospital. They included 26 male and 14 female. Their age range from 23 to 57 years old. As for control, 20 healthy individuals were collected, healthy person. 2 millimeter of venous blood were taken using 3 milliliter plastic disposable syringe, preserved in ETA tube, followed by gentle mixing in order to prevent blood clotting and store at -4 degrees. Then transported to the lab for DNA extraction. DNA extraction and quantitation. DNA extraction was achieved by using mini-prep system kit. Quantus fluorometer was used to detect the concentration of extracted DNA. Primer and probe preparation. Each lyophilized primer and probe was dissolved in free nucleus water to prepare the stock solution concentration of 100 picomole microliter. Then the working solution was prepared by adding 10 microliter of stock solution to 90 microliter of free nucleus water and stored at -20 degrees until use. The sequencing of primer and probe of 5-HTR1A as appear in this table. Genotyping using real-time PCR. DNA sample from schizophrenia patient, 40 patient, apparently healthy subject, 20 control, were genotyped for 5-HTR1A gene with the Tachman SNP genotyping assay using real-time thermocycler according to protocol recommended by manufacturer. Real-time PCR was performed in MIG-Q PCR cycler from Biomolecular System Australia. The component of reaction of a quantitative real-time PCR as present in this table. A master mix forward will reverse primer, probe, free nucleus water, and DNA. Thermal cycle protocol for real-time PCR program, 41 cycle, including initial denaturation, annealing, and extension with their times and temperature. Genotyping and allelic discrimination principle. The Tachman probe contain the receptor dye from and hex at the 5 end of a probe and the quencher dye at the 3 end of the probe. During the reaction, cleavage of a probe separates the receptor dye and the quencher dye, resulting in an increase in fluorescence of receptor. Accumulation of PCR product was detected directly by monitoring the increase of fluorescence of the receptor dye. When the probe was intact, the proximity of the receptor dye to the quencher dye result in the suppression of the receptor fluorescence. This picture shows the genotyping principle. Statistical analysis. Hartley Wimberling equilibrium calculated using a web tool. An odds ratio with the 95 confidence interval were also calculated. A two-tailed p-value was considered significant. Data were appeared as mean of standard deviation. Independent sample t-test was used to compare between means of studies group. Result. The study results were constructed by analyzing the information and data obtained from 60 plus samples, 40 patients of schizophrenia and 20 apparently healthy as control. The age of all subjects were a range of 19 to 57 years old as shown in table 1. This table shows the age, gender, and family history between the control and patient. Non-significant difference was observed between the patient and control. Molecular quantification of 5-HTR1A gene using real-time PCR melting curve of the 60 sample patient and control. Number and percentage frequency of 5-HTR1A gene genotype, and there are hardly one per occasion in control and patient. This genotype between the expected and observed show non-significant between patient, patient and control. Genotype and allele frequency of 5-HTR1 gene in control and patient group. These genotype between patient and control show non-significant differences. The genetic association with the demographic characteristic. Between age, we classify the genotype of schizophrenia patient to patient above 40 and under 40 years old, and the second table show the genotype without a limit for age. The results show also non-significant association between these three genotypes. In gender, genotype of schizophrenia patient also show non-significant association. Conclusion: the present study, to our knowledge is the first study to examine the association of 5-HTR1A gene with schizophrenia patient in a sample of Iraqi people. The RS-1042468 within 5-HTR1A gene is not associated with schizophrenia. This polymorphism is also not associated with age and gender in schizophrenia patient group. Recommendation: schizophrenia is multifactorial disease, and there are a number of genes associated with schizophrenia. Thus, direct sequencing of this candidate gene seems to be important promising approach such as NRG1, DTNB-B1, DAOA, and DISC1. Research on 5-HTR SNPs such as 6295 established their role and association with schizophrenia. Further studies are required to understand the serotonin system at the molecular level. Study the association of hormones such as estrogen, oxytocin, and testosterone with schizophrenia. Study the effect of vitamin D on schizophrenia. Thank you very much.
شكرا جزيلا ست نور.
شكرا لكم، سعادتكم راسي.
لا ما يضر شيء، بس احنا نفتح الان باب السؤال. خاف احد من اخوان الحضور عنده استفسار سؤال من الباحثة.
دكتور محمد تفضل.
السلام عليكم.
عليكم السلام.
اهلا دكتور.
شلونكم دكتور؟ شلون صحتك؟ ان شاء الله بخير.
يا هلا جميعا.
اشكر الباحثة على المحاضرة القيمة.
شكرا لك.
ممكن عندي سؤال اشوف what the relationship between the oxytocin and serotonin الربط بيناتهم، ايش الربط بين الاوكسيتوسين، هرمون الاوكسيتوسين والثيرويدين اللي هو هرمون السعاده الاوكسيتوسين، بس الربط ايش الربط بيناتهم؟
هو صراحة أنا من كنت أسرج على هذا الموضوع كان أكو إنه علاقة بين الهرمونات الثلاثة، بس يعني كربط بيناتهم دول إنه عليهم علاقة ويا الشيزوفرينيا، يعني إنه من يقل المستوى مالتهم حي يعني شلون نقول إنه يصير بهم أعراض شيزوفرينيا، لأنه هذا الأوكس مثل ما قلت هسه الأوكسيتوسين إنه له علاقة بالسعادة، السيروتونين أيضا هو هرمون عادة، فيعني إنه
الصوت اختفى دكتور احمد؟
الصوت ما ادري، هي يمكن من يم ست نور.
يبدو لي من ست نور. نستميح دكتور محمد عذرا لانه انقطع الصوت للباحثه. ننتقل الى الباحثه الاخرى دكتور زياد، وعلى امل انه ترجع لنا ست نور في وقت قصير. دكتور زياد.
لحظه دكتور، لحظه بس لحظه دكتور.
نعم.
عندنا نرجع معتز محمد، ما ادري موجود او لا. عندنا ضمياء، دكتوره ضمياء مكي حمزه، وعندنا علاء سعد طالب موجودين. اخواني اللي موجود منهم اخواني اللي بهاي الجلسه خلي يسوي ريز هاند. و عندنا زينب خضير عفوا، سيف طالب، جاسم
موجود. عندنا مروان ميثم دكتور زياد. مروان ميثم. نعم
إيه
مروان ميثم.
تفضل استاذ مروان.
بهاي الجلسه دكتور مروان. احنا عندنا دكتور علي الدليمي اذا موجود.
فتح الدكتور مروان. مشكل.
يلا ما مشكل.
الو السلام عليكم.
عليكم السلام ورحمه الله، مرحبا بك.
السلام
مروان.
My name is Marwan from بصرة. جدا شاكر مشاركتكم ومشاركتي وياكم. إذا كان الصوت واضح؟
نعم الصوت واضح.
اوكي، اقدر اسوي شير للبرزنتيشن دكتور؟
Go on. Okay, go on.
اوكي.
واضحة دكتور.
اوكي، السلام عليكم.
واضحة. السلام.
عليكم السلام.
Shall I speak in English or in Arabic? What you recommended?
اوكي دكتور، باللغة مال البحث نفسها عادي.
Okay. My name is Marwan Maythem. I'm from Basra. This is the title of my research: is the prevalence of beta-lactamase CMY2 gene and quinolines genes QRA, QRP, and QRS that are isolated from Salmonella species from human, animal, and milk. Okay. First, we talk about our introduction of our research about Salmonella. Salmonella has remained the potential threat for many diseases like typhoid fever, like enteric fever. We can transport from food to human. We can transport from environment to the human by contaminated food, and also the food that are from animal origins like milk, like meat, like any milk products like cheese or ice cream maybe, or yogurt. And also we have a huge problem with this kind of bacteria, like other bacteria, that is resistant to the mechanisms of antibiotic resistance. So in this study, we will focus about identification the salmonella, the bacteria, the salmonella from the samples, and also check the antibiotic sensitivity and searching for the genes that are responsible for antibiotic sensitivity and located their genes, the locations of the genes. Is there in the chromosomal or in the plasmid or whatever? The reasons, how can bacteria take these genes and become more resistant or multi-resistant for antibiotics? Materials and method. In present work, we undertaken the isolate and identification of the salmonella by using the biochemical, the old school, let's say the old school method, using the biochemical identification for the bacteria, and also use a serological identification for testing. Sorry, this is another sound. صوت آخر أكمل له فقط.
بس هذا من هذا جبته، لا من جبته هذا عرفه. اي عطاله ما
Sorry.
تفضل دكتور تفضل سكت.
Okay, thank you. So we use a biochemical test for identification of this bacteria in this study, and also use a serological testing and confirming the identification by using molecular detection by using polymerase chain reaction. And also we extraction the plasmid, searching for the genes that are responsible for antibiotic or antimicrobial resistance. And also we do a little experiment called plasmid curing by specific procedures that make the bacteria lose the ability to activate their plasmid or to eliminate the effect of the genes that located in the plasmid, and see the responsibility of the resistance of the antimicrobial is from the chromosomes or from plasmid. The total samples were 287 samples were collected from milk. We have two types of milk, directly from animals and indirect, that from markets. And also we use a human source as a samples, like feces, like the hands of the milkers, and also from the stool and basracity. And this is the types of the primers we used. We used the first primers, the 16 RNA that primer used for identification the salmonella, and also we use the quinolines, QNRA, QNRP, and QNS. This is the genes that are responsible for quinolines resistance, and also we use the beta-lactamase genes build CMOI2 that's responsible for beta-lactamase or beta-lactams resistance, antimicrobial resistance. The results. The over identification of the salmonella was 9.7% from total samples that using the biochemical test, using the API2 systems, and also using serological method. While when you use molecular detection, we have 6.1%. That means that molecular detection that minimize or lowering the over ratio of detection. It means that might be other bacteria is the phenotypic is resemblable for the salmonella, but this is not the salmonella according to the 16 RNA gene that used for identification. And also, we found out the highly resistance of the salmonella was against the chloromethylene, vancomycin, lincomycin, and rifampicin by 100%, where ciprofloxacin was sensitive. All the bacteria, the salmonella bacteria, was 0%. Okay. The plasmid curing, we use a temperature plasmid curing, and also when you curing the plasmid and recultification of the bacteria in the media, we saw that the bacteria lose their ability to resistance the ampicillin amoxicillin, and azithromycin, streptomycin, and other chlorphenicals. That means that the plasmid has the major responsibility for resistance for beta-lactamase antibiotics. And also, that is confirmed when we extraction the plasmid and do the molecular detection, the presence of the specific gene, that the BLCMY gene that's responsible for beta-lactamase resistance and equinoir genes, we saw the matching between the curing results and also the PCR results. We found in the PCR results that the gene beta-lactamase CMY genes high was a high percentage in the plasmid, whereas the bacteria are sensitive to quinoline antibiotic, and there is no bands for quinoline genes that quinoline P1RA, quinoline B, and quinoline S. And this, the first photo, see the PCR amplification by using 16 RNA gene, and was the size of the gene, the 16 RNA gene was 550 base pair, 5 base pair. While this is the show, the figure 2 that show the differences between wild type and the cured type. This is the wild type, where the bacteria was resistant to many antibiotics. But when you cure it, use the plasmid to cure it, we will notice that the bacteria has those ability to resistance the same antibiotic with the same concentration we used it before. And also, this is the photo that showed that the beta-lactamase bands, beta-lactamase G, BLC, CMY2 genes, the bands, we found it in many results that confirm that ability of the salmonella to resistance, the beta-lactamase resistance, the beta-lactamase antibiotic, the antibiotic resistance. And this is the photo that showed that amplification of the gene that extracted from a plasmid by using specific kit, using for plasmid extractions, and use the specific primer for amplification, the genes that we extracted from the plasmid. And conclusion, the beta-lactamase CMY2 gene that related of the presence of the transposomal elements responsible for mobilizing an equine. That means that there is a transposomal gene can be jumped from plasmid to the chromosomal or otherwise from chromosomal to plasmid that may make the bacteria have the ability to resistance to the beta-lactam antibiotic. While there is no quinoir genes located in the plasmid, that means that no resistance against the beta-lactamase, the quinoline antimicrobial. And the plasmid has the major favor for the bacteria to protect her against the antimicrobial or antibiotics. And we also, when we use a plasmid curing method, we show that the bacteria lose their ability to resistance. We use the plasmid curing depend on the simple method. This is curing plasmid, named the plasmid curing by temperature. It's increasing the temperature, incubation temperature with a specific media, and the results show that the bacteria lose their ability to, for example, express from the gene on the plasmid, or cannot maybe transfer this gene from the plasmid to the chromosomal. So the high temperature will minimize the ability of the genes that are located in the plasmid. And thank you for giving me this opportunity to share my research with you.
Thank you very much, Dr. Marwan. You're welcome.
Thank you.
طبعا مقاومة المضادات الحيوية واحد من المواضيع المهمة جدا في صميم اهتمام أهل الأحياء المجهرية. إخواني نفتح الآن باب الأسئلة إذا عندكم أحد عنده سؤال. اللي عنده سؤال يقدر يسوي ريز هاند على مود نعطيه مجال.
دكتور طه، دكتور زياد، دكتور طه يبدو عنده استفسار.
دكتور ده شوف تقدر تسوي لي انميوت من يمك هذا.
السلام عليكم.
اهلا دكتور. مرحبا دكتور محمد.
سؤال والله للباحث. معكم الاستاذ الدكتور محمد خالد شندالة، جامعة الموصل، كلية الطب البيطري.
اهلا وسهلا دكتور.
بس عندي ممكن يعني سؤال للباحث. what's your opinion about the main cause for this resistance؟ المين causes ايش يعني انت حسب توقعه المين causes for the spread this resistance؟
Okay, thank you دكتور. The main causes of resistance بالمناسبة هي resistance تصير عندنا مشاكل كلش عظيمة، وخاصة احنا هاي الأيام جاي نواجهها. احنا من مثلا يجي بيشنت لأي مختبر ويسوي وذل تيست ويلقى الوذل بوزيتيف، الأطباء راح يسوي له recommendation for many drugs, many antibiotics. هاي الانتيبايوتكس من ياخذها المريض راح ما يشوف أي هيلينج أو أي يعني استجابة إليه، لأن للأسف السالمون اللي صار اعتبرت حاليا XDR اللي هو Extended Drug Resistance، يعني صارت يعني ماكو اي انتيبايوتكس، او مو نقدر نقول ماكو اي انتيبايوتكس، واي انتيبايوتكس صارت يعني ما راح تؤدي وظيفتها وتقتل هاي البكتيريا. هي عده اسباب او عده تفسيرات علميه لهاي الريزيستنس. انا اتخذت جانب كلش مهم هو البلازميد، دور البلازميد حول انتقال الانتيبايوتيك ريزيستنت جينز ما بين الكروموسوم ما بين البلازميد وما بين بكتيريا وبكتيريا ثانيه عن طريق انتقال البلازميد. فكان الفوكس في الدراسه مالتنا هو حول البلازميد، وقدرنا احنا نسوي تجربه معينه ان هو نقلل من شلون ما نقول من البلازميد activity or plasmid expression by doing the plasmid column temperature. لقينا تغير كلش قوي بالاستجابه حول او العفو للريزيستنس للانتيبايوتيك، وكانت استجابتها للانتيبايوتيك اكثر، يعني كانت ريزيستنس كانت سنسيتيف. بس صراحه دكتور احنا سويناها على اساس الكيربي باور اللي هو انهبيشن زون، لكن هي البحوث الحديثه كلها تقول انت لازم تسويها على المايكرو برود ديليوشن. فيعني ممكن البحث باكر عقبه ناخذه يعني فكره بلازميد كيورينج والانتي بايوتيك تيستينج باي ام اي سي، لان هي اقرب تكون الفايفو اقرب من ما تكون ان فيترو.
انه استفسار استاذ مروان. استاذ مروان.
نعم اسف.
يعني بلازميد كيورينج انتم ضمن البروسيجر، يعني هل استخدمتوا البلازميد كيورينج باستخدام الحراره؟ يعني هدفكم كان بلازميد كيورينج برفع التمبريتشر؟
اي دكتور، صراحه احنا ردنا نسوي بلازميد كيورينج باستخدام مواد اخرى مثل الاس دي اس فور اكزامبل، او يمكن اتصور كان اكو فشي الاكريدين اورنج، هاي فد ماده ممكن تضيفها للميديا وسبيسيفيك كونسنتريشن هم اتصور راح يسوي بلازميد كيورينج. بس تعرف وضع كان يعني وقت البحث وضع العراق كان مو تمام للاسف، وان شاء الله سوى بلدنا افضل، فاحنا اتجهنا بس للتمبريتشر ونخلي فد ميديا خاصه بيها موجوده.
نعم هو الاكريدين اورنج.
اي ونسوي يعني انكوبيشن، اختارنا درجه حراره 39.
هو الاكريدين اورنج يستخدم للكيورينج ايجنتس، يعني اكريدين اورنج
اس دي اس انا اتصور يمكن قاري هالريسيرش
اس دي اس. بس دائما نصيحه علميه، ابتعدوا عن الكريدين اورنج او اثيديوم برومايد. ليش؟ لان هاي يسمونها ميوت. لا بالاضافه لهذا هي ميوتاجينيك، لانه احتمال يصير لكم طفره في الكروموسوم، تغيير صفات البكتيريا. فدائما يفضل استخدام الاس دي اس او سلفونيك اسيد. السلفونيك اسيد ولهذا بالادويه يستخدمون السلفونيك اسيد شركات ادويه. ليش؟ لانه من تاخذ الانتيبايوتيك وتاخذ وياه سلفونيك اسيد، هذا يعمل ككيورينج ايجنت، يعمل على فقدان، لانه راح يلتصق بمناطق او يرتبط بمناطق الجدار، لانه الخليه من تنقسم مثل ما تعرف راح البلازميد يلتصق بالجدار حتى يتنصف، يعني يتضاعف ويتنصف. فهاي المواد تمنع التصاق البلازميد بالجدار عفوا غشاء البكتيريا الداخلي، وبالتالي تفقد الصفه. وكل طريقه اخرى يعني التطفيل بترانسبوزون، يعني هو هذين غالبيه الانتيبايوتيك ريزيستنس هي جايه من ترانسبوزونات مثل ما تعرف.
صح.
اكو طريقه اخرى، هاي طريقه كلش ممتازه. ليش هاي طريقه ممتازه؟ لانه اذا استخدمت الاكريدين استاذ مروان واستخدمت اي ماده اخرى، هاي راندوم عشوائيه ما نعرف بها ممكن راح تشتغل. لكن اذا استخدمت الترانسبوزون هاي سايت سبيسيفيك ريكومبينيشن راح يرتبط بمكان محدد، وهاي صفه irreversible. يعني من ينضرب عندك الترانسبوزون يدخل في منطقه الترانسبوزون، هاي راح تكون irreversible وتفقد الصفه المقاومه البكتيريه نهائيا، تفقدها البكتيريا كصفه مقاومه. يعني هاي من باب الاضافه للبحث. بحثك ممتاز، بحثك رائع،
اشكرك
وان شاء
دكتور،
الله
اشكرك.
يفتح. وموفق استاذ مروان، موفق انت ومجموعه
الله يحفظك.
الباحثين. الله يسلمكم.
اشكرك
شكرا
دكتور.
جزيلا. اشكرك.
شكرا لك وشكرا على مداخلتك. احنا نستفاد من عندكم، انتم اساتذتنا، واكيد يعني هاي المواضيع احنا مهما سوينا سيرش بالبيبر، بس اصحاب الاختصاص واصحاب اللي شلون ما نقول النوت العلمي على استناد صحيح مثل ما تفضلت حضرتك. يعني انا لحسن الحظ انا مسجل هذا الكلام كله لان اريد اراجع له، ممكن باكر عقبه مثلا يعني اريد اسوي ريسيرش، اسوي فوكس على هاي القضايا، وهم اذكرها ضمن الدسكشن. يعني جدا شاكر الاضافه مالتك دكتور.
شكرا جزيلا.
بالتوفيق ان شاء الله استاذ مروان، بالتوفيق ان شاء الله، موفقين جميعا. شكرا
توفيق ان
دكتور
شاء الله.
زياد.
طبعا دكتور
زياد.
عفوا تعقيبا ايضا على سؤال دكتور محمد من جامعه الموصل ايضا. طبعا هي البكتيريا باستمرار تحاول تطور امكانياتها يا دكتور على مود تقاوم الادويه والمضادات الحياتيه وتزيد من يعني فرص بقائها بالبيئه والطبيعه. احنا يعني لحد الان هذا البحث هو ترند، يعني جزء من البحوث اللي تتابع هاي العمليه شلون البكتيريا. انا دكتور مروان اذا تبقى على الجلسات باكر ان شاء الله هم عندنا بحث على السالمونيلا، بس احنا شغلنا على الفيج، على البكتيريوفاج.
بكتيريوفاج.
نعم البكتيريوفاج ايضا تستخدم البكتيريا يعني improve the genetic information that enable these bacteria to resist antibiotics. أولا شكرا جزيلا. اخواني بعد منو عنده سؤال اذا هذا يسوي ريز هاند. طيب
نعم دكتور زياد.
اول اجل دواء زياد اجل. دكتور محمد اذا تسمعني عفوا. اخوان دكتور علي.
نعم.
انا دكتور دا اشوف الاسماء من موجود من الاخوان، اتمنى يرفع ايده. دكتور ضمياء، دكتور معتز، دكتور علاء سعد،
دكتور علي الدليمي موجود.
علي الدليمي موجود قبل شوي شفته. نعم. موجود بالجلسه. اتمنى
رفع
العفو دكتوره زينب خضر موجوده هم دزت لنا البرزنتيشن.
دكتور علي الدليمي هسه ودكتوره زينب وراه
ان شاء الله.
دكتور علي تفضل. دكتور زياد
اي دكتور
لكم الفضاء
بس خل اسوي
كوهوست. نعم.
مو انا
دكتور احمد الربيعي ان امكن
خلي يسوي كوهوست.
دكتور احمد الربيعي، دكتور علي يمكن تحول الى كوهوست لطفا.
نورنا دكتور علي، هو يسمعنا هسه.
دكتور اعتقد دكتور مروان عارض
اوكي هسه
مشكله.
بس دكتور علي اصر يعني انه يشارك بالمؤتمر ويتابع الموضوع جدا يعني.
اهلا وسهلا به ومرحبا. دكتور احمد الربيعي ممكن تحول دكتور علي الدليمي الى
كوهوست.
اي دكتور احمد الربيعي
حولت دكتور.
شكرا جزيلا دكتور احمد، شكرا جزيلا لسه الاجابه. شكرا.
دكتور علي. ما ادري يسمعنا دكتور علي او لا، ما اعرف.
دكتور علي بس الميكروفون لو فتحه انا هساعده. دكتور علي اسمعنا هسه.
اهلا وسهلا
جزيلا.
اهلا، سمعك هسه.
اهلا بيك. دكتور تسمعني؟
نسمعك دكتور، تفضل. سوي شيء.
اقدر اسوي شير؟
اي تقدر دكتور، تفضل، سوينا كو هوست. طلعت الشاشه.
دكتور ظهر شيء يم حضراتكم؟
نعم واضحه دكتور علي، الدريمه واضحه. دكتور علي واضحه. First international virtual binding of RGD peptide on gold nanoparticles for novel human carcinoma cells targeting. تمام؟ هو هذا موجود عندنا.
بالضبط.
اي.
الحضور الكرام الباحثين الاساتذه الاعزاء جميع القائمين على المؤتمر العلمي الدولي، السلام عليكم ورحمه الله وبركاته. Today I would like to talk about binding of RGD peptide on gold nanoparticle for novel human carcinoma cell targeting. I am Ali Al-Duraimi, PhD student in Department of Molecular and Medical Biotechnology, College of Biotechnology, Al Nahrain University. Introduction. Cancer is a major public health problem worldwide. More than 11 million people are identified with cancer every year. It is estimated that this number will increase to 16 million by current year. This means that the current treatments are ineffective in treating cancer. The conventional therapies such as chemotherapy, radiation, and combination with surgery are not sufficiently effective to further distribution and non-specific delivery of chemotherapeutic drugs. Nanotechnology-based diagnostic and therapeutic systems have supplied a potential option for most cancer treatments. Among all nanoparticles, gold nanoparticles are desirable candidates due to their individual properties, including chemical resistivity, enzymatic stability, low toxicity, biocompatibility, specific optical properties, surface modification. It can easy to conjugate with image probe and targeting agents such as antibody and aptamer. Delivering sufficient concentration of therapeutic to most cancer cells without producing off-target side effect against health cell remain problem. One of the choice to resolve this problem by use the cell penetration peptide as RGD, arginine glycine aspartic acid. RGD sequence is derived from fibrinoctine, a broadly distributed protein in the extracellular matrix of cells. Because of this, cell can recognize the RGD group and form a localized docking adhesion point on extracellular matrix. Cancer cells which boast overexpression of RGD receptor can therefore highly targeted by the side. Breast cancer continue to be most the frequent cancer and death among women. Aim of this study to enhance anti-cancer efficiency. We produce a cancer therapeutic conjugate consists of gold nanoparticle and RGD peptide and test its impacts against human breast cancer cell line MCF7 using various parameters. Methodology. First is the preparation of gold nanoparticle by straight reduction method. Then the gold nanoparticle functionalized with RGD peptide to form gold nanoparticle RGD. Then characterization of gold nanoparticle and gold nanoparticle RGD using UV visible spectroscopy, FTIR, FESEM, and transmission electron microscope TEM. Then in vitro assay using antioxidant assay of gold nanoparticle and gold nanoparticle RGD using DPPH free radical scavenging activity. Then cellular targeting assay to detect the measurement of cellular uptake activity of gold nanoparticle RGD and compare with gold nanoparticle alone. Viability assay using anti-proliferative assay for MCF7 cell line as MTT assay and use clonogenicity assay for MCF7 cell line. Apoptosis detection using cell cycle analysis. Result: the synthesis of gold nanoparticle and gold nanoparticle RGD. Gold A is gold salt. Gold salt in yellow color contain a gold ion Au plus. This gold salt incubation with sodium citrate as reducing agent, sodium citrate, and boiling convert the gold salt to gold nanoparticle in the wine red. Gold nanoparticle have a negative charge. The size of gold nanoparticle depends on the ratio between the citrate and gold salt. Gold nanoparticle have negative charge on their surface. These can lead to aggregation with each other by the time. To overcome this problem, CAP with 2020 to distribution and prevent aggregation. Then functionalize with SHRGD peptide to form gold nanoparticle RGD. Characterization using UV visible spectroscopy. As shown in the figure, the red line is gold nanoparticle peak 520. When put this peak number in formula, specific formula, using that produce the diameter of gold nanoparticle around 20 nanometer. As shown in the green line, the gold nanoparticle RGD shifting the peak from 520 to 530. This may be due to the increase in the particle size when functionalized with RGD peptide. On the other hand, the absorbance also increased from 3.5 to 4.4. When the particle size increased, absorbance also increased. Characterization by FTIR. This FTIR assay used to detect the functional group in the compound. As shown in figure, the red line is the gold nanoparticle and the green line the gold nanoparticle RGD. The functional group is in the gold nanoparticle is same in the gold nanoparticle and RGD, but we found two functional group present in the gold nanoparticle RGD but don't present in the gold nanoparticle alone. This compared the functionalized gold nanoparticle with RGD peptide. Characterization by FESEM. As shown in figure FESEM, the gold nanoparticle with spherical size arrange average size about 20 nanometer. Gold nanoparticle RGD shift the size increase to average 30 nanometer. Electra confirm the size using the transmission electron microscope TEM. As shown TEM, the distribution of nanoparticle. The shape of nanoparticle is spherical. The size of gold nanoparticle about 20 nanometer. Gold nanoparticle RGD show the spherical, but some particle show the another form as triangular or hexagonal. Due to the functionalized RGD to the nanoparticle can shifting the form, change the form of nanoparticle, slight change in form. And the size also increase about 30 nanometer. Antioxidant activity of gold nanoparticle and gold nanoparticle RGD. Compare with vitamin C as a positive control. Gold nanoparticle antioxidant activity about 60%, but gold nanoparticle RGD about 83%. This means the gold nanoparticle RGD have a hydrogen atom that donate to the free radical to reject it to radical DDPHH convert from purple to colorless. This means the product of gold nanoparticle it has antioxidant activity. Measurement of cellular uptake. Measurement of cellular uptake activity as shown in the figure. Untreated cell, the cell stain with Dabi stain. Gold nanoparticles show non-homogenized around the cells. Gold nanoparticle RGD show aggregated, highly aggregated around the cells due to the cells has overexpression of RGD receptor. This cell overexpression of RGD receptor when attacked by RGD, gold nanoparticle RGD, this can block these cell and cause growth inhibition. Gold nanoparticle functionalize with gold nanoparticle RGD to form gold nanoparticle RGD. This cell membrane of healthy cell normal expression of RGD receptor as shown in figure, but cell membrane of cancer cell with overexpression of receptor. This conjugate combine to cancer cell only and cause a block the cell and finally cause the inhibition, growth inhibition of cancer cells. Use MTT assay show on the red line the gold nanoparticle and the green line the gold nanoparticle RGD. As show gold nanoparticle can decrease the cell viability about 40%, but gold nanoparticle RGD can decrease the cell viability more than 60%. On the other hand, when used the normal cell line, the slight effect of gold nanoparticle on normal cell line about less than 10%. Other assay used to test is clonogenicity assay. Clonogenicity assay, it is assay a clonogenicity used to detect the antiproliferative effect of cell to form colony, ability of cell to form colony. Untreated cell to high ability to form colony, gold nanoparticle slight effect of colony, but gold nanoparticle RGD has high effect of colony formation. The gold nanoparticle RGD have long period stability, may be used in long-term drug therapy for cancer cells. We use cell cycle distribution, MCF7. This assay used to detect the percentage of cell population in each phase, G1, S, and G2 phase. As shown in this figure, the percentage of cell in G1 the significant decrease from 51% to 48%. About S did not change, but G2M have significant increase from 26 to 28. This statistical analysis of different phase the cell population in this assay. This means the gold nanoparticle RGD can arrest the cell cycle in G2M phase and can cause the cell inhibition or cell death by apoptosis. Conclusion, The RGD peptide can be conjugated to gold nanoparticle by direct cross-link using a simple methods. Functionalization of gold nanoparticle with RGD peptide can be used targeting cancer cell and overcome the low cellular permeability of encapsulated agent. The gold nanoparticle RGD have highly cytotoxicity toward breast cancer cell. In contrast, they have low cytotoxicity toward normal cells. Finally, we suppose that our study will provide an innovative idea for the single-step synthesis of other nanoparticle and multifunctional use for tumor imaging or therapy. Thank you for good attention.
Thank you so much, Dr. Ali,
Thank you,
for
Dr.
this great presentation. Now we are opening the discussion. Please, anyone have a question or inquiry, you have just to raise hand, please. اخوان اللي عنده استفسار بس مجرد يرفع ايده. اوكي، يعني يبدو انه ما عندنا سؤال الان. حسب ما اعرف الدكتور سيف موجود، رجاء يرفع ايده على مود ننطي صلاحيه انه يلقي خلاصه شغله. دكتور سيف حسب ما عرفت موجود. دكتور سيف بس ارفع، بس ارفع ايدك.
ارفع ايده. نعم نعم. دكتور سيف اللي بعده دكتور وفاء، خلينا نشوفها. دكتور وفاء ويانا؟
دكتور الان دكتوره زينب دزت لنا تسجيل كامل للبرزنتيشن مالتها.
ماكو مشكله. اهلا وسهلا.
يتفضل دكتور.
السلام عليكم. السلام عليكم اخوان.
عليكم السلام. عليكم السلام دكتور، مرحبا بك.
طبعا دكتوره زينب من جامعه بابل كليه طب الاسنان تعتذر بسبب الانترنت ورسلت لنا باوربوينت صوتي، ان شاء الله راح نعرضه لكم.
دكتور علاء. دكتور علاء.
نعم دكتور.
ممكن نشارك وياه الصوت اثناء ما نشارك الباوربوينت؟ هاي من الشير نفسه اكو علامه صح للشير مال الساوند فور كمبيوتر. عيد المشاركه، عيد المشاركه من الجرين اختار شير ساوند وذ ساوند كمبيوتر.
اوكي دكتور، بس اكو مشكله دقائق.
قول لهم شو ادري.
الصوت
introduction.
طبعا.
Natural herbal
دكتور زياد.
elements and spices have been utilized
موجود
as
الصوت؟
food additives, flavoring agent, and preservatives for hundreds of years. In addition, they have been used for medical purposes. The increased exposure to antibiotic during eradication of bacterial infection has led to the development of resistant strain of bacteria, not only for certain antibiotic, but also there may be multi-drug resistant.
شنو دكتور علاء توقف الفيديو؟ دكتور علاء تسمعني؟
نعم دكتور اسمعك، بس اذا يتوقف منه حول على السلايد الثاني.
خاف اوتو، شغله اوتو ذاتي. دكتور احمد اذا ساعدنا بي بلا زحمه.
دكتور اعتقد المشكله بالانترنت.
دكتور روح على سلايد شو بلا زحمه عليك.
العفو دكتور، بالانترنت المشكله، المايكروسوفت يوقف ويرجع مره ثانيه.
اعتقد ممكن عاد خلينا نحول على غيره ونرجع نعاد جبل ما.
اوكي استاذ سيف اذا موجود، دكتور سيف. حسب ما اعرف الدكتور سيف ايضا موجود. استاذ سيف اذا موجود رجاء ريز هاند.
نعم دكتور بدأ العرض يمه.
The study aimed to evaluate the inhibitory effects of the essential oil from cloves, thyme, and cinnamon compared with ampicillin on some oral bacteria, include Staphylococcus aureus, Streptococcus mutans, Lactobacillus species, Pseudomonas aeruginosa. Materials and methods. Herbal and spices essential oils. Herbal and spices essential oils were selected from blue
دكتور علاء. توقف البث دكتور علاء. يلا اوكي. دكتور احمد تسمعني؟ دكتور علاء مطفي الصوت ترى ما نسمع شيء.
دكتور زياد.
نعم دكتور احمد.
اعرف منو اللي بعده يعني حتى نشغل الغرفه.
اي ما ادري هسه انا دكتور سيف اذا موجود، لان حسب ما فهمت الدكتور وفاء يعني دكتور سيف طالب جاسم يسوي
ما
ريز هاند
هو
رجاء.
يرفع مثل يسوي
زين
ريز
طيب.
هاند. وسويت
زين
سيرش على اسمه
بحضر.
ما لقيته.
هي يا ابني علاء سعد، علاء سعد العتابي، نور قاسم كمل، الدكتور ضمياء مكي حمزه، هي ولا علي وخالد خليل، دكتور معتز ايضا، معتز محمد الطائي.
الو دكتور زياد مرحبا.
نعم دكتور وفاء تفضلي.
حبيت مداخله انه استاذ سيف قال دقائق وانا ان شاء الله اكون متحضر دقائق. استاذ سيف.
مشكلة اي من الموجودين بالجلسه هسه حاليا يعني ممكن يسوي له ريز هاند خلي يتاكد من وجودهم.
عادي
اي شخص من
اخواني الموجودين في هذه الجلسه حاليا حاضرين هم موجودين، بس خلي يسوون ريز هاند على مود يعني المشاركين قصدي في بحوث او على مود نقدر نقدم السيره مالته.
الو
دكتور وفاء تفضلي. يا اللهم صل على محمد.
نعم دكتور زياد وياك. الو
نعم دكتوره ما منتظر احنا استاذ سيف ما عندنا.
منتظرين
راح هسه نفوت شوي على فاصل مؤقتا بين ما يجهز
اوكي دكتور. اوكي
استاذ احمد لا تسمعني شوي فاصل بسيط بين ما يجي استاذ سيف يجهز لنا الباوربوينت مالته.
Words stand mute to describe its development and scientific status. It becomes, through its achievements, an eminent scientific edifice. That is University of Al-Qadisiyah. The university is situated in the city center of Diwaniyah Governorate, about 180 kilometers south of Baghdad. University of Al-Qadisiyah is established in 1987, Faculty of Administration and Economics and Faculty of Education. Currently, it includes 18 faculties. Along the previous years, the university has established specialized and scientific faculties to provide society with accredited research and scientific services which introduce academic outcomes to the market.
السلام عليكم جميعا، ونرحب بالسادة الباحثين والمشاركين والداعمين لمؤتمرنا الدولي الافتراضي الأول في التقانات الإحيائية، وتحت شعار ببحثنا العلمي نواجه جائحة كورونا المستجدة، حيث المناخ العلمي لتبادل الأفكار ونتائج البحوث العلمية الرصينة لخدمة المجتمعات. أهلا وسهلا بكم في هذا المحفل العلمي البهي، وشكرا لكم جميعا. محمد عبد الوهاب العسكري، عميد كلية التقنيات الإحيائية في جامعة القادسية. السلام عليكم. We welcome the researchers, participants and supporters virtual international conference of biotechnology under the slogan Our Scientific Research Challenges the COVID-19 Pandemic, to exchange the ideas and scientific research results to serve the societies. Once again, you are most welcome in this gorgeous scientific forum. Thank you. Mohammed Abdul Wahab Al-Askari, Dean of the College of Biotechnology, University of Al-Qadisiyah.
الشمال الغالي يبقى شامخ عالي يسمو كالجبالي كاكا تمام والوسط قريبة والغربية حبيبة والفرح نجيبه باسم العراق
اوكي دكتور احمد تسمعني؟ تسمعني دكتور احمد؟ استاذ سيف اذا توفرت رجاء ريز هاند، ارفع ايدك. استاذ سيف الاخ سيف طالب وجاسم فخري احمد اذا موجودين رجاء علمونا بوجودكم عن طريق الريز هاند.
موجود هذا سيف؟ غير موجود.
دكتور محمد عزيزي يبدو انه دكتور سيف ما اعتقد يعني. تفضل دكتور يلا. هذا استاذ سيف يلا. استاذ سيف عيني. استاذ احمد اجى استاذ سيف، هذا نسوي ريز هاند، رجاء نسوي كو هوست على مود يعرض. استاذ احمد وياي؟
استاذ احمد؟
استاذ احمد وياي؟
استاذ احمد اذا ممكن تحوله الى استاذ سيف الى كوهوست.
نعم دكتور وياك.
استاذ احمد سيف تحول الى كوهوست، وعلاء كامل ايضا تحول الى كوهوست، يمكن اثنيناتهم. سيف رافع ايده هسه. استاذ سيف تفضل افتح الميكروفون. سيف طالب جاسم. استاذ احمد عيني. استاذ احمد.
نعم دكتور وياك.
سيف طالب جاسم، سيف طالب جاسم موجود امامنا هسه، اذا يمكن تعطيه كو هوست وتفتح الميكروفون.
استاذ سيف تقدر هسه ان شاء الله تبلش تعرض.
موجود سيف طالب جاسم امامي.
ويسمعنا؟ ما ادري دكتور.
اي يسمعنا. سيف طالب موجود امامي هسه كو هوست. خليه يفتح الميكروفون.
بس هو ما فاتح الميكروفون. حاول
انا سافتح لي اياه. استاذ سيف وافق على فتح الميكروفون. استاذ سيف تسمعني؟
دكتور يمكن طلع ما دا يشوف.
لا موجود. الو عليكم السلام.
الو دكتور تسمعني عيني؟
اسمعك واضح. يا اهلا وسهلا بيك، شرفتنا. تفضل على العرض البرزنتيشن.
لي الشرف دكتور بكم، وتمنيات ان شاء الله بالموفقيه لمؤتمركم ان شاء الله تعالى، النجاح والتميز ان شاء الله تعالى.
شكرا 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 شكرا. استاذ سيف تفضل شير اللي عندك، المحتوى اللي عندك.
دكتور راح ما عندي محتوى صراحه، بس مجرد راح القي خلاصه البحث اذا تسمح لي.
تفضل تفضل.
راح مجرد القاء خلاصه البحث. بحثنا عيني دكتور، بحثنا بعنوان the study the effect of diamino acid and nanoparticle on biofilm candida albicans. هذا البحث طبعا بصورة عامة كانت الهدف منه إيجاد علاجات بديلة لغير من المضادات الفطرية. فاستخدمنا خلال هذا البحث نوعين من الأحماض الأمينية التابعة للداي أمينو، ولاحظنا أن الداي أمينو ذات تأثير عالي على تثبيط النوع من أنواع الفطريات، وهي الكانديدا البيكانس المصيبة للجلد وخاصة في الإنسان. لكن هاي الداي أمينو أسيد كان تأثيرها بصورة فريدة من نوعها، طبعا تأثير عالي أكثر من ما تم دمجها مع جزيئات النانو بارتيكل. الأسباب عديدة، لأن من أسباب التأثير تثبيط أو دمج الدايو أمينو أسيد مع النانو بارتيكل راح يؤثر كل منهما يؤثر على ميكانيزمية العمل لدى دخوله على الكانديدا البيكانس، من ناحية تأثيره على السيل وول، من ناحية تأثيره على البيبتيدوغلايكان المكونة جدار الفطريات الجلدية وغيرها من المسببات الأخرى. لكن طريقة العمل بصورة أساسية أخذنا الكانديدا البيكانس، وطبعا هاي الكانديدا البيكانس تم الحصول عليها من بصورة أساسية من الجلد في إحدى مستشفيات بغداد، وتم تنميتها على وسط السبرايد ديستروز أجر بدرجة حرارة 37 درجة سيليزية لمدة 24 إلى 48 ساعة. بعد ذلك تم قياس الMIC لكل من الأحماض الأمينية الداي ألينين والداي بارولين بطريقة التشو كلتشر بليت. ولاحظنا أن الMIC للداي ألينين هو تقريبا ما يقارب ال 25 مايكروغرام على المل، وايضا الدايبرولين على لاحظنا ان 100 مايكروغرام على المل. بعد ذلك تم اخذ هذين الحامضين الامينين وتم تعريضهما بصوره مباشره ضمن وسط زرعي لتنميه الفطر اللي هو كانديدا البيكان. ولاحظنا من خلال عمليات التثبيط ان الداي اليلين والدايبرولين كان لهما تاثير كبير جدا على نمو الفطريات، وهذا ما يدل على استخدام هذا الفطر مستقبلا كعلاج بديل عن المضادات الفطريه بصوره اساسيه. لكن طرحت لنا فكره دمج هاي الاحماض الامينيه مع الجزيئات النانو بارتيكل لمعرفه تطور اكثر خلال العلاجات. لكن لاحظنا ان دمج هذين النوعين اللي هي الداي امينو اسيد والنانو بارتيكل، لاحظنا التاثير كان اقل من ما هو يعني نريد نعرف ان نحصل على نتيجه افضل. لكن النتيجه كانت صراحه مو ذات افضليه كبيره ما حصلنا عليه من تاثير كل من الحامضين الامينيين اللي هو الداي امينو الينين والداي بارولين. وبالتالي لاحظنا ان الداي بارولين كان له تاثير عالي جدا على الكانديدا البيكان بصوره اساسيه، وهذا طبعا اعطانا فكره كاستخدامها كعلاج بديل عن المضادات الفطريه اللي استخدمناها سابقا، اللي هي سواء كانت النستاتين او غيرها من المضادات الفطريه الاخرى. ولاحظنا ايضا من استخدام السيلفر والليثيوم اللي هي تابعه لجزيئات الاحماض الامينيه، لاحظنا ان هاي الاحماض الامينيه عند دمجها مع السيلفر والليثيوم، وكانت تراكيز السيلفر والليثيوم اللي هي 25 مايكروغرام على الميل، بينما الليثيوم كان 12.5 مايكروغرام على الميل. لكن هذه التأثيرات ايضا انطت فعاليه، لكن ليست بفعاليه الاحماض الامينيه عند استخدامها بصوره مفرده دون دمجها مع النانو بارتيكل. بصوره مختصره توجيهنا الاساسي لهذا البحث هو ان يكون بحثنا استخدام علاجات بديله، وهذه العلاجات البديله صراحه نوعا ما تكون غير مكلفه وغير مكلفه للطالب وغير مكلفه للمؤسسات العلميه، من ماده الى استخدامنا لهذه الاحماض الامينيه اللي هي اساسا تكون متوفره بصوره كبيره ضمن المختبرات ومتوفره ضمن المخازن عديده في كليات التربيه وغيرها من كليات العلوم الاحياء. بالتالي تسهل فرصه للطالب لاستخدامها مستقبلا كعلاجات وزياده الدراسه عليها، دمجها مع انواع اخرى من الاحماض الامينيه، دمجها مع انواع اخرى من الجزيئات النانو بارتيكل، ليست الليثيوم او السيلفر جزيئات الفضه، او دمجها مع مكونات كيميائيه اخرى، وبالتالي راح نحصل ان شاء الله على طريقه فعاله لعلاج هذا الفطر الذي يسبب الكثير من المشاكل الصحيه للانسان ولغيره من الكائنات الحيه الاخرى. طبعا استاذ احمد هذا مختصر البحث مالتنا، واحنا نتشكركم على الاستماع والتواصل معنا. الو دكتور احمد؟
نعم
تسمعني دكتور احمد؟
شكرا جزيلا استاذ سيف.
سمعك دكتور، بس يحولين دكتور زياد. دكتور زياد؟
عيني تفضل استاذ احمد. تفضل وياك استاذ احمد.
دكتور انهي التقديم.
اوكي.
نعرض. العفو دكتور زياد، نعرض.
اي دكتور علاء.
عندك دكتور علاء جاهز؟ المحاضره مال دكتور
موجود
جاهزة؟
موجود، بس نشوف الانترنت ما اعرف. يلا هسه
أوكي.
نشوف.
حاول.
Method: in vitro assessment of antibacterial activity using agar well diffusion assay. The agar well diffusion technique was applied for the detection of antibacterial action of both herbs, essential oil, and antibiotic. Swab from bacterial growth was inoculated into liquid media and incubated at 37 centigrade for 18 hours. Normal saline was used for diluting bacterial suspension. The suspension turbidity was adjusted to be comparable with standard tube Macfarlane 0.5 to get an average suspension containing 1.5 multiplying by 10 exponential 8 colony forming unit per mol. Molar Hinton agar was streaked by swab of bacteria suspension according to Kirby power techniques. Essential wells were created on the culture media, then 0.1 mol of herbal essential oil was added to wells. They were incubated at 37 centigrade for 24 hours. All readings were measured in triplicate and zone of inhibition were measured as
دكتور علاء.
دكتور علاء
دكتور علاء.
أنه ايضا المشكله ما زالت قائمه.
مو مشكله دكتور زياد. نعم الفضاء لكم دكتور زياد.
دكتور انا اشوف انه
دكتور زياد. نعم
يعني
نعم.
عفوا دكتور، اكو مشكله بالباوربوينت. نعتذر يعني بالنيابه عن دكتوره زينب.
ماشي. شكرا جزيلا اخوان الاعزاء الكرام على هذا اليوم الحافل بين الجلستين الصباحيه والمسائيه في مؤتمرنا، مؤتمر الافتراض الاول لكليه التقنيات الاحيائيه. الجهود مشكوره للاخوان في اللجان التحضيريه والعلميه طبعا جزيل الشكر والتقدير للسيد رئيس جامعة قادسية والسيد عميد كلية التقنيات الأحيائية للجهود وللتوجيه وللدعم. وحاليا هو موجود ويانا السيد العميد، يعني كان متواضع للغاية وشفتوا حتى مداخلاته. الآن أترك المايك لدكتور محمد العسكري ليدلي بدلوه. شكرا.
شكرا استاذ دكتور زياد على هذه الكلمات الجميله الموفقه، بارك الله بك. اتقدم الشكر الجزيل في هذا اليوم الاول، نهايه اليوم الاول من مؤتمركم، مؤتمرنا جميعا، مؤتمر كليه التقنيات الاحيائيه، بالشكر الجزيل الى السيد رئيس جامعه القادسيه المحترم، الساده المساعدين واللجان التحضيريه، والجندي المجهول الذي يجلس الان خلف الحاسبه في بغداد ويدير هذه المنصه الالكترونيه ببراعه وبمهنيه ممتازه وعاليه، الدكتور أحمد الربيعي. كما تقدم بالشكر الجزيل إلى كل اللجان والسادة مدراء الجلسات ومقرريها اليوم، وكل من شاركنا من كي سبيكرز، كينوت سبيكرز والمتحدثين. اليوم كان يوم حافل بالإنجازات العلمية، كان يوم مليء بهذه الطروحات الجميلة والنتائج والخبرات التي تم استعراضها من الخبرات الموجودة داخل العراق وخارج العراق في الدول العربية والدول الأجنبية. في نهاية هذا اليوم أتقدم بالشكر الجزيل لكم جميعا الحاضرين معنا ومن غادرنا، ونلتقي على بركة الله بكم غدا الساعة 10 صباحا لاستكمال باقي جلسات المؤتمر، حيث غدا سوف تكون اليوم الأخير بجلستين وذكر التوصيات. شكرا لكم جميعا. الفضاء لكم دكتور زياد.
شكرا جزيلا دكتور، تحياتنا للجميع، وإن شاء الله نكون استفادينا من هذا الحضور العلمي. طبعا احنا نباوع الأسماء قامات علمية معروفة جدا يعني بالجامعات العراقية تشاركنا الآن. احنا سعيدين بوجودكم وإن شاء الله الإفادة للجميع. أترك الأثير الدكتور علاء، الدكتور أحمد اللي الجندي المجهول مثل ما سمع دكتور محمد، أن شوية ينطينا فاصل. شكرا.
شكرا دكتور زياد، وشكرا دكتور محمد العسكري على هذه الكلمات الرائعة. إن شاء الله هذا المقطع الفيديو نهاية الجلسة. شكرا جزيلا.
السلام عليكم جميعا، ونرحب بالسادة الباحثين والمشاركين والداعمين لمؤتمرنا الدولي الافتراضي الأول في التقنيات الإحيائية، وتحت شعار ببحثنا العلمي نواجه جائحة كورونا المستجدة، حيث المناخ العلمي لتبادل الأفكار ونتائج البحوث العلمية الرصينة لخدمة المجتمعات. أهلا وسهلا بكم في هذا المحفل العلمي البهي، وشكرا لكم جميعا. محمد عبد الوهاب العسكري، عميد كلية التقنيات الإحيائية في جامعة القادسية. السلام عليكم. We welcome the researchers, participants and supporters of our first virtual international conference of biotechnology under the slogan "Our scientific research challenges the COVID-19 pandemic," to exchange the ideas and scientific research results to serve the societies. Once again, you are most welcome in this gorgeous scientific forum. Thank you. Muhammad Abdul Wahab Al-Askari, Dean of the College of Biotechnology, University of Al-Qadisiyah.